So we've reached the bottom at 1,758 meters, and already you can see that we have largely rocky bottom, small patches of sediment, and you know, this little feature that we've landed on is just loaded with some of the corals that I was expecting to see. So this is certainly a good sign.
And so what we're looking at has actually been pretty indicative of what we've seen before on some of these dives on the tops of these normal faults. So we see kind of highly broken, brecciated, and kind of rubbly fragments of pillow basalt that have been either cemented together by biogenic sediments or just kind of through the shearing force of this normal faulting. And you see some small intact primary pillow basalts. Whenever you see those kind of lobate features, and a really sheer face.
Hello, pilot. Fantastic. It's looking good. Looking good start. Why don't we begin by taking our second Niskin sample, and right here in front of some coral and a fish, so probably yield some good DNA. Thank you. This looks like a grenadier, what I used to call the rat tails. Kind of a pointed nose. You can see the little barbel sticking under the...
Let him go.
Thanks.
Get a mobile. Go to mobile critter.
I had some very interesting email from Ken Sulak about the fish yesterday, and I will relay some of that information the next time we get a good look at fish face.
I got you. Your Jim turned down pretty hard. Roland, are we talking a decade today or not?
No.
Okay.
We fixed the issue, so normal operations.
You want minimum 15 today?
For Delta?
Yeah.
Depends on the situation. This looks perfect. Whatever.
So, pilot, I'm seeing lots of stuff here on the rock to the left, and so maybe we could just kind of sit here for a little bit and get a view on several of the different species so we know what we're looking at.
Copy. I'm going to go for this cluster over here.
Sounds good.
Or lasers or aiming.
I'm almost overwhelmed, so I'm glad you're making the choice.
Copy. Yeah, you want to come in partial and try to get like this group here. Looks like we've got a nice variety here. Partial.
Yeah, so the one just above the lasers on the left, kind of, how would I describe that color? Kind of beigey pink, is a Chrysogorgia, family Chrysogorgiidae, and a very common deep sea family. And the depth that we're at today, I'm really expecting to see a lot of these Chrysogorgiids, primnoids, and keratoisidids, really common and really diverse down here. The lasers are now passing over a stony coral. I should have said the Chrysogorgia is an octocoral. That's a stony coral, probably Lophelia. I see a white sponge to its left, and I notice a hint of bright purple just on the lower part of that Lophelia colony. That would be an encrusting, yeah, you can see it down there, an encrusting octocoral. Has this beautiful color.
Oh, lights. Yeah, I do.
Yeah, we're getting a nice close look at it here. You can see the Lophelia in the foreground. There we go. This kind of grows as a mat or a ribbon over other substrates. So here it's overgrowing dead parts of the Lophelia. And you can see the Lophelia is actually providing a substrate to a number of other organisms. I'm seeing sponges. I'm not sure if that branching white structure was a branching sponge or a hydrocoral. I think it was a sponge. hydrozoans, cup corals. I would like a close view when you're done with this of that white whip.
Okay.
Thank you.
Stand by.
The yellow fan that you see above the white whip that we'll zoom in on in just a moment is a classic Paramaricia. That's the genus Paramaricia, classic sea fan or gorgonian in the family Plexauridae. And very often, many of the species that we see have an association with brittle stars, and I can see one there wrapped around, its arms wrapped tightly around the branches.
Go for the base of the white whip.
Okay, so what I want to see, is this a bamboo coral or primnoid coral? I believe it's a primnoid coming in, but maybe not. Oh, look at the spininess on this. So just slow down there a moment, Scott. Huh.
Got more zoom video.
It's max zoom.
Max zoom. Oh.
Yeah, let's move up a little bit. I'm now beginning to think that this is a bamboo coral, and I'm saying that in part because of the density of the polyps and the long needle-like white structures that you see arising from it. Wow. Interesting.
You want to
Very
get any closer to it, or
if you can?
okay,
I am pretty close to it.
Okay.
as close as you can get.
Yeah.
Yeah. So this must be a bamboo coral, a keratoicidid. And
Going up, see if we can find
I'm
it.
not sure I recognize it. Unfortunately, my
Obvious
colleague Les
point.
Watling is not in the room today. He's otherwise occupied.
Keep it on my lower swing arm.
But this is a pretty
Can
good
stow it if you
view.
want.
I can
Or
even
just a little
see
bit.
what's going
See
on
that?
in the,
Yep.
the polyp walls. I haven't seen a single node. Interesting.
The upper's clear.
If it wasn't for the spikiness, I felt I saw more spikiness down below than up here.
How's that?
I would
Going
say
all
that
the
this
way?
is
Oh, that's fine.
a
Do you agree that the upper's
primnoid.
clear?
What I'm trying to do right now, I'm sort of straining my eyes, is to see, can I see
Almost to the end there.
scales going up the polyp
Keep
body
that.
wall?
Yeah, it's a
I
tough
kinda
angle
think
for me to see the.
That
Wide
I
angle
do.
does
I'll
look
bet
good,
the
yeah.
spikiness that I was noting below is just all of the tentacles.
Is that really max zoom
And
video?
these
That is
polyps
max
do
zoom,
appear
huh?
to be arranged in what we call
Now, once we
whorls,
finish it, I
so
can
kind of like
pull out and
a
push back
circle
in.
of polyps going around the colony. So, yeah, that's a
Hold that.
primnoid octocoral. Thank you. Excellent.
Out, in.
Thanks.
I'd like to show science something I don't think they recognize. It's below the bottle brush orange one. Very small and wispy.
I'm going to go for the...
Yeah,
gold one.
I think it was a hydrozoan fan, a hydroid. It's growing off of the Lophiolae colony, Roland.
No, I think it was a different coral, one of those very wispy corals that you like to see.
Let's go for the gold one, Roland. Yeah.
Yeah, so this is a nice view of the Paramaricia with its associated brittle star, ophiuroid. We've seen lots of ophiuroids. The brittle star is one of the members of the Echinodermata, are really common in the deep sea, and they live in a lot of different kinds
Try
of
to hold this
habitats, and some of them
Beauty.
associate very closely with corals. And so the one that you're looking at here would essentially probably never live directly on the rock seafloor. It would spend its entire life associated with coral, and probably its entire life associated with this
Copilot,
one
no, it's
coral.
halfway through the
As far as
shifting.
we understand, it's not feeding on the coral. Instead, it's feeding on things that are landing on the coral. That's great. Beautiful shot. I'm also
Be
seeing
advised your light
tucked
bank,
into some of the polyps.
feedback.
There's
Yeah, I saw that.
amphipod
So,
crustaceans
if you
on
need
the
updates,
far
I'll let you
right.
over
There's a
here on my
small
GUI.
mysid crustacean that's a
It just
possum
didn't
shrimp.
get that GUI didn't get rebooted.
Understood.
Celeste Watling has arrived in the chat room, and he says, he reminds me the genus name for that brittle star is Astroschema, and he agrees with me that that whip we were looking at is a primnoid, maybe the genus Convexella. Thank you. It's kind of tricky, you know,
Yeah.
these whip-like corals. There are lots of different kinds of corals. Oh, let's zoom back in to the right of that clump. There's a fan that's sort of pointing towards the right side of the screen, almost at the bottom of the screen right now. So
Yellow
bottom left of the
lower right.
lasers, about 5 o'clock to the lasers.
Copy. The
Now it's directly right of the lasers.
Got it. Okay, let me move over a little bit. Stand by one. Talk about the later yellow colony.
That's what I'm talking about.
All right. We got to maneuver one sec.
So now in this view, how many different species do you think you see?
One,
So
two,
clear on
three,
the
four,
upper.
5, 6, 7, 8, 9,
Okay.
10, 11, 12, 13 corals easily, plus lots of sponges
talk
and
about
all
this
the
one, right?
other stuff that's associated with them. So this is an Acanthogorgia, different family from all of the other corals that we've looked at so far. It's another kind of octocoral.
Really, I still
A
have the
family
plugs
is the level above
Yeah, that's
genus,
good.
which is
Good
above
lighting there.
species.
Copy. Once I take off, we can turn them off.
And so we can recognize this by the relative height of the polyps relative to the thickness of the tissue and the fact that the polyps wouldn't retract totally into the tissue. So this is a good observation. Genus Acanthogorgia, family Acanthogorgiidae.
Pull to full.
Okay.
Well known for their, quotes, crown of thorns, sclerites, or spines that are wrapped around the top of the polyp. All right, what else to look at? There's so much to see.
Still a coral I'd like to show. him
Lophelia.
lower left
There's a
of
cup
the other
coral,
one.
that bright white
We're
thing
looking at.
just above
Tilt down.
the
Go for that
Acanthogorgia
Right,
we're
right,
looking at.
right. Sorry.
Now what you're saying, is this pretty indicative of what we've seen on the other dives in general? In terms
No,
of-
so this is really characteristic of this depth range across the Atlantic and even the Pacific. And the species may change, but the genera and the families are the same. But we really
Try to
haven't
hold that
been in
line.
this
I was
zone yet
rotating
so
on you,
far in
and-
this expedition.
Very good.
So
Flip.
I think we're seeing lots of different
Nothing
species.
in the pink.
That cup coral is dead. We're just seeing a skeleton. I'm not sure what the pinkish stuff is on it, but if we can go up just a little bit, pilot, above this, I think I saw a live one.
Oh,
There's
Where
one of
did I
those
see
wispy
it?
zoom out
ones
so I can
at
find
the
it.
bottom of the screen.
There it is.
Oh, the one with the
that-
flesh on it?
Exactly.
The pink,
kind of pinky brown.
transparent.
Yep. So that's a nice
Connular
view
shape.
of the cup coral from the side. So a cup coral is basically a solitary version of this brownish coral that you're seeing in
Take
front of
more.
it, the Lophelia. They're both
Yeah.
stony corals, so they both belong to the Scleractinia. They both secrete this very hard
Ron, I'll try
calcium
to
carbonate
level you out
skeleton.
here.
But the stony coral, excuse me, the solitary, let me try again. The cup coral is solitary. And so that's just one polyp, one really large polyp with its own
See the
private
corporate-
skeleton.
Chris?
Whereas if you
Yes.
focus
Oh,
on the
okay.
foreground in this
It's like,
Lophelia, you see that there are all this branching, and there's
check my
lots of
social
these
page
little
for me. Make
cups
sure they're
that
happy.
have a deeper orange
You see
color.
there's very
Each of
small
those
white
is a polyp, and it's just
right
that right
near the
now the
bottom
tentacles are
edge.
retracted in it. So that's a
Just right
colonial type of coral. The other one's a
very
solitary.
small.
Great, good
coral.
stuff. Thank you.
It's Scott. Okay, video. We come wide. No, I didn't see it. Video. We'll come wide. I think they're.
That sort of fuzzy colony that's just
works.
above the
They haven't
Lophelia,
yet
that's
seen
another
that.
kind of
That's the
bamboo
one I'm
coral.
still
That's
trying to get
Acanella.
them to see.
And yesterday on our
Right
dive,
above
we
the
did
right
see
laser
lots and lots
point.
of Acanella. They were one of the dominant colonies that we were seeing. They're the ones I said had the base that could be on a rock or in the sediment.
Okay, I'm gonna take off.
It's possible that this could be the same species as that. I'm not 100% certain, but there is one species that's really widespread, has a wide depth range. How about we go to the right?
Got a lot to see here.
Yes, indeed. It's going to be tough to tear away.
Okay.
Manuela Ramos on shore is saying the Scleractinian
Hey,
looks
you
like
want me to keep the fogs
Solenismilla variabilis instead of Lophelia.
Yeah,
so I
I
was
think we're
referring
still in close. Let's
referring
just
to it
keep
as
going for now.
Lophelia.
Thank you.
She's suggesting it's a different
You
genus.
want me to stow your
Same basic
port
structure
side
of
doors?
stony
Yeah, bring
coral
it
that's
to
branching.
match that one. Yeah.
Understood.
And thank you for that, Manuela. I'm really not that good at identifying them. How about that brushy brown thing that's branching on the left side of the lasers?
Large branching. Copy.
Yep. So this is a black coral. I'm actually seeing two different kinds of black corals. There's an orange whip-like one behind it to the right, but this branching one,
Come in, Marshall.
I think this looks to me like a branching, what's called a parantopathes.
Hold that.
And I see that this one has several squat lobsters on it. You can see 1, 2,
Take it
3,
easy,
4,
so look
5,
at the
6,
brow.
7,
Understood.
8,
So,
9, 10. That's very interesting. Yeah, if we can get a good view of any one of those. So very often we'll see a parantopathes, and I'm used to seeing the ones that don't branch. So this is a black coral, and they
Come
will
out,
often
video. I'm
have
gonna
two
your
crabs
position.
associated with them, a male and a female. So
What are
here I'm
those?
seeing a highly branched
Hold
colony, and there's looks like there's
Hold
a
that.
dozen
Yeah.
or more of these squat
I'm
lobsters
gonna
on
kind
there.
of pirouette to those.
So that's
One
quite
of those red
fascinating.
bulbulous things are.
Yeah, what are those? Yeah. Like almost like little honeycombs, like little holes in them.
It's got sponge shape, but I haven't seen that before.
I'm going to go for my left toe, and then I'm going to go for a close-up of this subject.
Brow looks good.
Copy. All right. Theo, come in. Let's go for a squat lobster on branching. Hold that. Wait for interest.
Yeah, great view. So these squat lobsters, chirostylid, that name translates to Latin as referring to those pointy, long arms, chelate clawed arms that they have.
Okay, so
And they have specially modified back legs that allow them to get a good grip on these corals. And certainly in some of the studies that have been done, just like the brittle star that I was describing earlier, there's a very clear associate-host relationship, meaning some of these crab species, the species is only found on one species of coral. And I don't know that that's the case for what we're seeing here, but it may be the case. What's really interesting about that from an evolutionary perspective and from a conservation perspective is that protecting the coral protects all the associates with it. Or you could say if we lost this coral, we would lose all of the species that depend on it.
I'll try to pirouette, Roland.
Nice view. Another thing you can see on this black coral is the polyps themselves are extremely small compared to some of the other black corals.
Understood. Yep.
Zoom out a little bit.
Said several times they get their name from the skeleton, which is largely protein-based, and it's deposited in layers. And
2.71
very dense,
nautical
very
miles,
strong. It's quite hard to break.
Next video.
A lot of strength to it, I guess tensile strength.
Sounds good. Thank you much.
Snap this one.
So, pilot, I am getting some questions about whether this is a stable enough position to make some collections.
Yeah.
Okay.
You come out video. You come out video. Thanks.
And we'll have to stand by because I've got so many requests that we're going to have to sort them out. Maybe we could have a look at that orange whip while we sort out exactly what's the priority here.
Understood. This one up here?
Yes, indeed, right in the center. Thank you.
All right. I'll come back in for it. Video? Yeah, hold that. I'm going to get closer video.
What's your brow? Looks good on the port side.
That could be good.
Yep.
It's going to be busy.
Not steady. Steady. Okay, video. You come in. I'm pretty steady here.
Okay, so we're looking at another black coral here, and this one you see has much thicker, fleshier polyps that are arranged just on one side of this branch. Another thing I'm seeing, in addition to being called the black corals, some of these they're also referred to as a group as the thorny corals, and that's because the skeleton has a variety of spines that arise from it. And I noticed if we pan upwards on this colony, you can see the bumps on the back side of those polyps, and you can see those little black dots. That's perfect. Look at that focus. Awesome. The black dots are the tips of the spines that are arising from the skeleton. And so when the tissue's off and you rub your fingers along it, it can be really abrasive.
They look fine.
I know, but and it's quite remarkable how much diversity there is in the shape of those spines and the size of those spines. And so a lot of the black coral taxonomy, especially in the early history of the taxonomy, was completely based on those spines and the shape of those spines. And that is in part because if you collect these by dredging, as they did back in the 1800s, the tissue is often stripped off, and so you only have the hard parts left to look at. This is probably in the relatively new genus Aphanosticopathes. I think it was only described this year or maybe late last year. It used to be called Sticopathes, and we've determined that evolutionarily it's related to a different group.
And can you explain to me what sticopathes or what was
Aphanosticopathes?
Aphanosticopathes means?
So I don't remember what the stico translates as. The pathes, these all belong to the order Antipatheria. So the order Antipatheria are the black corals. And that name translates to against disease. And it goes back to Aristotle's days, the ancient Greeks. And they believed that if you were to grind the skeleton, the powder would be useful to combat diseases. And so it got this name Antipatheria. And so traditionally now, most of the genera that are named have the name Pathes associated with them. So it's a matter of sticko. I'm not sure if that refers to a stick. That seems
like
too
that.
simple.
Yeah.
But
So many things to choose from here.
yes, there are.
This is amazing.
All right, so maybe, let's see here. I've had a request about maybe the Paramercia, the Primnoid whip, the Acanthogorgiid. I'm seeing right in front of me the Acanthogorgia. So next to that curly orange whip, you see there's a fan just to its right? That yellow fan?
This one?
Yeah.
Looks like there's two here.
Yeah, how do you feel about that as a position? I mean, there's so much here. Let's choose something that's easy for you to sample, not difficult.
That one is not super easy.
Okay, so let's keep going.
Okay. There's definitely going to be easier ones than that one.
Sure.
Let's see.
Let's have a look at the fish while it's going by,
Understood.
since that's mobile. Thank you.
Video coming on the head. Hold that. Okay, come in further. Looks like it's a little bent there.
This one looks kind of beat up. Lots of little scars on its side.
It was rotated, yeah.
Yep.
I wonder if it bumps
You can
into
tell by
the coral.
Jumps in it.
Eel-like, I'm not sure if that's a
Extra
synaphobranchid eel.
Yeah, turn those off.
Understood. Fog's coming up.
Different kind of fish, just above center.
You want to take it? Snapfish, yeah.
Yeah, good. This looks like another grenadier. Maybe a smoother head than the previous one. It's actually taking us somewhere really interesting, and that's that yellowish blue coral that just bumped into. Yeah, so let's get a look at that next.
Copy.
Actually, no, I'm not so sure that's a grenadier. And just for people on shore, the coral, the bright white coral in the upper left corner just before we zoomed, that was an Anthothelidae, and we did collect an Anthothela on dive 1.
It's a good shot.
Let him go off. All right, video. Thanks. I'm going to go for the small one. Zoom out. I got to kind of figure out where that. There it is.
Yeah, so this is going to be in the family Plexoridae, which are, I would describe those as the closest to the sort of Gorgonian sea fans that you would see if you were snorkeling on a tropical shallow reef. The Paramercia that we looked at earlier, and there are two of them right here, that's the yellow fans, also belong to that same family. So I think this is going to be same family, different species. I suspect it's going to be a situation where we have sclerites, the hard parts of one color, and soft tissue of another color, which is why we're getting that sort of blue and yellow effect. Yeah, Mike agrees that was not a grenadier. Les Watling has an interesting analogy here to the problem that the scientists on shore are facing. And he says he thinks of this as the candy store dilemma. 4 kids go into a candy store where there are lots of candies, but they can each only buy one. They only know the candies they have seen. They don't know what else the store has. So how to determine what and when to buy the candy. And that's what we're facing with sampling.
The rock's a little crumbly here, but we'll get a view of it.
Turn on the extra lights.
Understood. Fog's coming up.
You come in, video.
That rock was just displaced. No, it's not enough of a rock. Understood.
Trying to get you as close as possible.
I would extend that analogy to say we're buying candy for all of mankind.
That's good. Yeah, you're right. Absolutely. Just the 4 kids have to decide what candy to buy for all mankind. So this is a really cool what we're looking at here. So the hard parts, the sclerites, and part of the tissue are yellow. And it looks like I'm seeing a blue pharynx. So the tube that you see inside of these polyps, that's basically the throat, the pharynx. And it is this really interesting blue color. But then the tentacles themselves are yellow in color. Oh, wait, but these ones are, Yeah, okay.
And we saw bicoloration in something similar to this yesterday, didn't we?
We did, but what we saw was the polyps were all one color, and the, let's say, the main tissue on the axis was a different color. But here I'm seeing,
Color you don't see very often.
yeah, the other view from the side, you could see the tentacles appeared yellow, the pharynx appears blue. So that means there's two different pigments, unless I was just seeing through. Yeah, this is a good one. I don't recall ever seeing this color combination. So I'm standing by, I'm looking in the chat room, waiting for someone. Oh, there, yeah, we have Asako is voting as this for a sample. Yep, Manuela says she's never seen this. So let's have this as our first sample, please.
Understood. Go back, can you help me?
It's certainly in the family Plexaurid. I have no problem saying that, but that's an extremely large family, lots of diversity. And at some point there was some discussion about natural products, and this group has some really interesting secondary metabolites. And I think I did note the other day that Plexaurids, Acanthogrogiids are a little bit more tricky to get the DNA out of, and it has something to do with the other metabolites that are in the tissue, maybe binding to the proteins. But
I imagine that's going to be a pretty tough problem to solve from an analytical side of just coming up with a new methodology to process that, given a really extremely rare sample size. You don't have a lot of sample to work with to develop those analytical techniques.
Yep. So you probably work on shallow water taxa to do that.
And after we finish this, if we can brush the lasers across the center of the branch there.
So some guidance pilot. This will be pretty flexible. It should be relatively easy to snip.
Okay.
We don't necessarily need any branch point, but I think it's going to be small enough that you'll have to do that. And then just for the audience watching, almost assuredly, once the manipulator touch it, you'll see the polyps retract, and this colony will probably turn into a yellow colony. We'll no longer see the blue tint.
Sweep the lasers across the branch.
That's my guess.
Scott, I'm thinking I'll probably cut the right branch off.
Yeah.
Once I get the manipulator close, so be a game time decision. But
yeah, I think that's just fine.
Video's clear.
Thank you.
Okay, you got hydraulics up.
Hydraulics are up.
All right. Keeping an eye on us, Lars.
I have good eyes.
All right.
Reasonable.
Arms coming on.
You want me to stay with this, or you want me to pull out?
Oh, it's perfect, Ron.
Okay.
Yeah, it's down there.
And for scale at home, those two red dots that you see in the rock
you
below
wanna
the
zoom in on the
colony are
polyps?
10 centimeters apart.
Polyp side.
I'm just touching him to see him react.
So you can see at least the tentacles folded. The polyps haven't yet pulled into the scenenchyme, but they should have the ability to fully retract into that yellow.
Okay.
The pilot's sort of tickling them. I'm actually surprised at the lack of response. Interesting.
Rotate over. Jaws. Is that the plan?
Asako Matsumoto, coral biologist in Japan, says that this is more reminiscent of a tropical shallow water coral than it is a deep sea coral, given its coloration.
Don't take it any lower.
This is what I was going to finish the thought earlier, is that the secondary metabolites I'm referring to are probably associated with these pigments. So the more colorful ones probably have more of these secondary metabolites.
Scott, is it okay if I get the entire colony?
Yeah, if it's small, yeah, just go for it.
Yeah, once you get the jaws next to it, get a better scale.
Looks like we'll
Did you
probably have
cut the
2
top?
large branches
Oh,
there.
yeah. Cut from the bottom.
Yep.
It's like I cut it right after that branching point. So I
Yep,
kinda
so
have
they'll
2
come off on
pieces.
2 separate
Yep.
pieces. Yep, that's fine. And I see there's also a hydroid colony growing in the center there. So now you can see most of the polyps have retracted.
Yeah, you can take
Some of the
some.
tips are out. There you go. Some are almost fully inside. Others look like little hemisphere domes.
Nice.
Very good, thank you.
Okay, if it didn't come out, I'm going to go for Stowe.
Let's just get a
Good,
couple
Rich.
seconds of that
Okay.
in case they want to edit with that.
Go ahead.
I'll hold that.
Discussing the closure.
Copy. 2.68.
And then 1 max tight.
255.8.
White
Maybe
lines that
reporting
you're seeing
outer.
are sclerites, I believe, which are calcium carbonate microskeletal elements that
here, I
are
think,
embedded
take
in
off.
the tissue. And
Okay,
with the
video's
polyp
clear.
having
Right.
pulled its tentacles inside, you're seeing the backs of them with
There we go.
those sclerites. And we use the
Leaving out
sclerites
heading
for
engaged.
the taxonomy, for the identification
Come off easy.
and description of these species. See a stock crinoid over there.
Let's see.
I'm
that's
almost
what
overwhelmed at the amount of stuff that's here.
Camera is full wide.
Copy. Thanks for that. Okay. Good for drawer.
Understood. Sure, coming up.
I might have to move the manipulator up a bit if the suction hose is going to strike, but see that?
Go.
Let's lose the extra lights.
Creeping in.
I'm watching the, see how close the wrist is to the top of the box?
Yeah.
So that's what I'm watching right now.
Start moving it.
Yeah. Okay.
I think you're on there. Good collection. Short coming in. Is it clear?
Yeah. Thanks.
Short in.
Oh, thanks.
Creeping in.
Yeah. Yeah. Good deal.
Hydraulic secure.
All right.
Where did it go?
Sample pilots.
Port outer.
Port outer, thank you.
And we're back.
Awesome, thank you. I'm
Oh,
still
big
trying to
C-star.
communicate some information. Yeah, go ahead with the C-star, and
Yeah.
then that red colony that was just below your lasers after that. Looks like a Paragorgia. It's
Copy.
just above the C-star, yep. The
Okay.
C-star may be feeding on this coral.
Let's see what I can do here.
Do you may want to see the lower section of it?
Let's see what I can do, Ron.
Go ahead, Survey. You're muted.
We're trying to look, look up at it.
Yeah, right.
Try to add that wonder factor.
Okay.
I called it Plexauridae purple polyps. PPP.
Purple polyp power.
Yeah.
Okay, come in. Hold that.
All right. Sorry, folks. I'm also typing notes to folks on shore, so there's multiple things to see here. So absolutely, this coral has climbed up on this, what I think Manuela identified as Selenosmilia,
There we go.
the stony coral. And the way that these asteroid sea stars feed is they evert their stomach from their mouth and lay it over.
There we go.
Yeah, what a fantastic, incredible view. So I don't see the stomach everted, but you can see the mouth there. There's all these
Turn them on.
Did you light that up, video?
little bumps
I don't think
and spines and
yeah,
things and
definitely
tube
helps.
feet
Good job.
clustered around that polyp. But it can evert its stomach, and
Auto depth got me.
oh, actually, I think it is.
I know,
It looks like
okay.
that
Oh, you
white
hit it?
frilly
Yeah.
stuff that
That's
you
what
see
it bounced.
just
Yeah.
amidst the
Hit.
pink and the orange.
you
I
hit
think
by
that's the
No, like
the pyloric
I saw it
stomach.
was an auto depth.
And
It's like,
basically,
so I punched
it
out.
everts
Yeah.
it
I knew that was
over
gonna happen.
whatever it is eating
Just
on,
got it
secretes
over with,
enzymes to essentially liquefy it, and then can transfer that into the gut. So has this
It's a nice
incredible power to feed in some unusual and
So every
rough
time I hit the
topography,
touch
let's
the
say.
rotator, that's
And
a
I'm
cut.
seeing on
So
the
I'm
right
gonna
side of
roll
the
it again
screen
for
here
the
3 of the
12,
tube feet
whatever.
that look
Yeah.
like they're attached to the
Roll it again.
closest polyp on this side. Interestingly, I know we're all focused on the sea star,
So you
but I
grab a hold
think
of
that
the
little
needle and
branching
eventually lock that
structure
in.
in the foreground right
Yeah, let me tell you
is
what they're like.
a small little bryozoan.
Copy.
We're zoomed in so much we don't usually get to see that stuff.
Okay, we can come out a little bit, see what else we see.
Les Watling suggests
full
that maybe
screen
it's
of
eating a
sea star.
soft coral that's
Copy.
growing on the
This,
Lophelia,
lighting.
but is certainly feeding.
Okay.
Yeah, so we're also interested in this red fan just behind there. And I'm
bubblegum.
trying to see, if that's a bubblegum. At first I thought maybe it was a Swiftia, but I think Pilot's absolutely correct. That is a Paragorgia. And I can see it also has its Astroschema brittle star wrapped around it. And we have another
you go
kind of
bubblegum?
Galathea crab
Okay,
crawling
bubblegum.
on the background there, having a look, saying, what's going on with this thing?
You want some attention too?
Yep,
He's got big
don't they all?
pincers
Very big arms.
relative to his body size.
Yep.
Maxine.
Yeah, it's a different kind of Galathea than the ones that you find on the
Hold
corals.
that.
So beautiful view here of a Paragorgia or Paragorgiidae, maybe I should say, because I'm not sure what genus it is, but it's in the family Paragorgiidae. These are the bubblegum corals. And you can see what look like wads of gum, which is how it got its name. In this case, the polyps are all retracted, so you don't see the tentacles out feeding. But they would normally be where you see those various lumps and bumps.
Full screen
One of the
bubblegum.
interesting things about this group of corals is they actually have 2 different kinds of polyps. So you may have noticed there's small wart-like structures as well. Those are called siphonozoids and don't have tentacles but do channel water in and out of the colony. Very good.
Beauty shot there.
Nice. Awesome. Next video.
Again, the Acanella bamboo coral
Coral
is
line.
what's filling up most of the right side of the screen. I see a couple of urchins here, lots more of that beautiful purple
Tilt down, put the
stoloniferous
corals.
octocoral. I see
Just show 'em
Bathypathes
what's up
black
here.
coral. And what's this large thing? Probably, oh, this is a bamboo coral for sure. I can see through the nodes on the main part of the stalk. Excuse me, I can see through the tissue on the main part of the stalk down there, the black nodes. So you can just see these black lines in between white, what we call internodes, the main part of the skeleton. So bamboo coral, and then lots of
Wanna try
really
something?
cool
Sure.
glass sponges up here.
Starboard lower. Deploy it a little bit.
Understood. Jocks on. Starboard lower coming out.
Just deploy a little bit. And swing it in. That's good. Swing in. Inboard. Easy inboard.
Inboard start.
Easy
Stand
inboard.
by.
Yeah.
Come back down a little bit.
You can watch it with the lasers if you want.
Yep. All right, coming
As you swing it in.
in. Inboard.
And then turn the starboard mains off.
Just trying to creep it in easy.
That's good. Turn starboard mains off. See if that looks like. Oh, probably light up more than that. It's fine. That's fine. You can turn it back on.
Starboard's back on. Get that back outboard.
Okay. Scott, was there something else here? What's the next subject you want me to zoom in closer
Just
on?
if you
The large one?
could get this large one,
All
if
right.
you get the planar view, just
Yeah, I'll hop up
rotate
there.
around a bit. Thank
Okay.
you.
All right.
And then we should probably get a good view of one of these sort of large platey fan-like sponges, the large white ones that we're seeing.
Okay.
I'm seeing several of them in this area, so it'd be a good idea to get a close look.
Copy.
Video, you want me to secure fogs?
Yes, please.
Rotator 16.
Toe. Port.
I'm clear now, aren't I?
And clear the lasers.
All right.
So I'm actually seeing several bamboo coral colonies here. As we were moving up, one just went off screen below. There's one to the right, and I suspect they're all the same as this one we're going to image nicely here in the center. You can notice that the main stem that's attaching to the rock is really quite thick. Can we get the lasers back on
Absolutely.
for a short period of time? Thank
Put
you.
right next to the base.
Yeah, thanks. There we go. So our 10 centimeters there. So I'm guessing the diameter of that skeleton is probably 2.5 centimeters. So that's pretty thick. They deposit their skeleton in layers. In some cases, they're annual layers. So I imagine this is
Are you a little
well
heavy
over
right now?
100
Pretty
years old,
neutral?
this coral.
Yeah.
A little light.
I accidentally pressed it in, so it disabled it momentarily. But yeah, it was a little late. Okay, video, you want to come in on the pops? Do it floating, so it'll be moving, so can't zoom in super far, but
That's your starboard toe.
So
Yeah.
we looked earlier at that whip-like coral that I couldn't decide if it was a bamboo coral or primnoid. Eventually we decided it was
Yeah,
primnoid, and
right.
one of the things I was looking for were
Thumbs up.
nodes. We've already seen nodes in this one down below in the skeleton, and this is beautiful lighting. You can see all these white lines in the polyp. So those again are the sclerites, calcium carbonate concretions that are secreted by the coral. The shape, the density, and the placement of those sclerites is one of the primary characteristics used to describe and identify bamboo corals. And so this unbelievably exquisite image that I'm getting gives me so much information. It's almost like having this under a microscope. Just fantastic. Look at that. Wow. And I do see that several of them have the prominent needles sticking in between the tentacles. Thank you. That was terrific.
thanks, video.
And the other part of this is where does the branch arise in this internodal brancher. So we have a lot of information there. Let's have a look at this sponge. Manuela is suggesting that this may be Faria herdendorfi. And if it
Full
is
wide view.
that
You want
species,
full wide?
this would be the first record in this area.
Bring it over.
Which, doesn't surprise me all that much, given
We'll just
that,
keep the lights
as far
on
as
for
we
now.
know, nobody's ever dove down here in this area. So a lot of this stuff is going to be new to hear. And as our colleagues, as Telma was noting earlier in the introduction, they really haven't had an opportunity to dive below about 1,000 meters. And so much of the stuff that we're seeing is all new. As we were swinging around here, I was kind of eyeing that bamboo coral that's just behind the one we were just imaging, and it looks like it crosses at the base. And I think it's
You want to give us
like
5 meters there?
anastomose to itself, meaning the two parts
Video
of the
come in.
colony have come back in contact and grown together.
Heading would you like to
So this is a glass sponge,
270.
and you can see it's very interesting sort of
285.
frilly appearance. It's growing in a sort of large
285.
paddle-like
5 meters,
shape, if you
285.
will. and it has a number of like protrusions or holes or tunnels.
I'm trying to bring it
Very
around.
often when we see
Just don't
these
want
kinds of glass corals,
contact
there are
this
many other invertebrates that are living associated with it. So it's very clear you can see the arms of these brittle stars that are on there. But often tucked into some of the openings are things like amphipods and shrimp and mysids, various kinds of polychaete worms. Right now, the only associates I'm seeing are the brittle stars.
Go ahead, Bridge. Yep, ready, go ahead.
And my daily reminder that sponges are mostly suspension feeders. And by mostly, I mean most of the diversity, most of the species that we know. Can we also, sorry, before we go, just to the left of that sponge, you see that stick that's kind of orange?
Sure.
So it's a crinoid, but the crinoid is being overgrown by something. And I've now seen 3 or 4 of these, so it might be nice just to photo document what's going on.
Copy.
Thank you. So yeah, sponges are suspension feeders, so they rely on channeling a current through the body and pulling out
Okay, video, come back in.
bacteria-sized particles for nutrition.
See if this toe lasts longer.
So what's going on here? Must be hydrozoans. Yeah, so what we're seeing here is a crinoid, a stalk crinoid, or a sea lily. That's an echinoderm, and that's what's produced this long stalk, and there you can see its arms up at the top. And the sort of fuzziness to those little side branches of the arms, those are the tube feet. So they're very, very small tube feet there, and the mouth would be just where all those arms come together. But then overgrowing it is another cnidarian. This is in the class Hydrozoa, and this looks like a colonial hydrozoan. It looks to me like what we call in shallow water snail fur. Hydroctinia, I believe, is the genus. Snail fur gets that name because it grows like this on the surface of a snail shell. And then each of those little upright stalks that you see is an individual polyp. It's got its tentacles, and that bright white spot that you saw at the tip, that's where the mouth would be. So very cool. Another example of how organisms that are growing up off the bottom are providing substrate for other kinds of organisms. Really increases the biodiversity of the community. The substrate here looks like a lot of old Selenosmilia, that stony coral rubble. So you have corals growing on older corals. All right, we can move on and find something else.
Onward and upward.
Oh, sorry, I had
Portman came
request for the Acanthogorgia. We did a zoom earlier on that, James. We got some actually really good footage.
Okay, yeah, we'd like to put in a ship move. Length 5 meters.
I think
Bearing
just below that pinkish fan, that's probably the anthothelia again. If we could just zoom in, you see the X formed by those 2 colonies there? I'm just trying to see if they're growing together. Thank you.
All good. The high flyer situation has been relayed to the
pilots. Yeah, so it does look like they have merged. It's not one in front of the other. Yep, that's exactly what's going on. So these 2 colonies have come in contact, and then the tissue, well, are they, or maybe they're just bumping into one another? It looks like there's one on the right and there's one on the left. Maybe they're not quite anastomosed. They're just sort of bumping and pushing, and then say, okay, I'll go left and you go right. There we go. Very cool. But it's the same. It's the same bamboo coral that we were just looking at a couple of minutes ago.
Okay.
Extra light's clear.
Copy.
Fog secure.
Let's see what the future has in store for us.
Yeah. Right in the center, you see that lone whip with the curl on the tip. So that's the primnoid whip that we imaged earlier. And the primnoidae, when they have a whip, very often it looks very smooth, and they often have that characteristic little hook at the top. So I like Les Watling's suggestion earlier of convexella. Plenty of the stony corals, these clumps of kind of light beige color, Selenosmilia. The more orangey bushes are the Acanella bamboos. Some yellow sponges, and then you're seeing a lot of rubble of these Selenosmilia corals, and that's what most of the black, very knobby tubular texture of the bottom is.
Go for the tube.
This is looks like a Euplectella sponge. Another kind of glass sponge. Forms a tube. I'm not sure that this one is all that healthy. Just based on the color, it's not really bright white, so I suspect it's just the dead skeleton there.
Okay, video, come out. Keep going.
On the left of it, the whitish stuff is an Anthothela, and on the right is a very young bamboo coral that's just starting to grow. Several cup corals. How about the far right? You see the red polyps that are extending off of the rubble?
Copy.
Just want to see if that's a young Paragorgia or if that's an Anthomastus.
Okay. Come in.
Go ahead, Bridge.
Yeah, so that's going to be Anthomastus or one of its close relatives. It's sort of commonly referred to as a mushroom coral. So although it has the same color as that bubblegum coral that we saw earlier, it has far larger polyps and it never branches. It just creates basically like a little globe on a stalk that the polyps emerge from. Thank you, pilot.
No. Okay, coming up.
The upper right corner, that large fan that you see there is a different kind of black coral. I believe that's a Storopathes. And then coming into frame on the right, that's another kind of black coral. That's a Leopathes, genus Leopathes. That's the one we saw so much of yesterday up on the shallower dive. They're really common. We made a collection of one. We got actually a nice big chunk of that, trying to determine if it was Leopathes expansa or Leopathes glaberrima. Again, seeing in the far left the Paramercia fan coral.
Let's take this black coral video. Just take a for confirm identity.
Yeah, so I'll wait to see our colleagues on shore to get information, but I'm pretty sure that this is Storopathes. And I'm basing that on the fact that you've got the main stems. You can see they're nice and black. In fact, that stem in the uppermost one, the leftmost one, you can see how shiny black it is, and that's because the tissue has been removed, and you can see the branches coming up above it where the tissue actually is still alive. So you can see why they're called black coral, even though the tissue may have a lot of different color. But anyway, the branches that come off the main stem sub-branch many times, and that creates this fan-shaped structure, and that's a characteristic of the Storopathes. And I am hearing that our onshore expert Tina in all things black coral agrees with me. Thank you, Asako, for passing on that information. So again, there's a marvelous diversity of things. There's almost so much to look at that it's hard to choose what to see. So what I'm trying to do is, as we go over, identify the things that we sort of grossly know what they are, and then we'll go in and get some zooms. But as we want to continue to move because we're interested to know, well, how far does this extend? And what I'm seeing right now almost blows my mind how much I see out into the distance. What a great view. There's a really large colony in the background, a couple of them.
You want the fish?
So I think we got, yes please, I think we got bang on another spectacular site that's going to have a very rich, high density, high diversity coral community. This is an Oreo, or I think they also call them Alfonsino, is that correct? Or am I mixing that up? I'm mixing that up. That's the barracks. This is Oreo Neositis. And I forget the species name.
60x9, nice.
And we saw a lot of these as well yesterday when we were up at about 1,000 meters. So clearly they have a depth range that extends from about 1,000 meters down here to right now we're at 1,748 meters.
Hit that sweet spot.
Yeah. What's he doing down there? Hey, little guy.
Sideways.
All right, we'll let him go.
Coming up with you.
Okay. You might let him go. But
so maybe we could do two things as we start to move up slope. Just left of center, there's kind of open branching whitish. I said left and I meant right, of course. That won't be the last time.
Copy.
Thank you. That larger one. So I suspect this is bamboo coral. And what I'm interested in is looking at this, and then we're going to look at the whip that's up the top left and
Gotcha.
see if we can see that they're different at the polyp level.
Understood.
Thank you.
You want another 5 minutes?
Neositis
Yes, I do.
All right, now,
helgi.
let's do another
Thank you,
5 minutes.
Carlos, for that
285, please.
name. That was for the fish that we just saw, the Oreo. So I believe this open branching
Wanna get close to
bushy
video?
colony is going to be
Bridge
another
opening.
bamboo
Get you as
coral.
much detail as possible.
Thank you.
Tracking
We'll
iris.
just get
Yeah,
a
we'd like to
close
put
look.
another
And
ship move,
again,
please.
one of the things we
Let me know when
concentrate
you're ready
on
to
is where do the
Range,
branches
5
arise.
meters.
So we want to see
Hydroid
are the branches coming from a dark spot, the node, which is protein, or are they coming from a white spot? And then we want to look at the polyps and see what can we learn about the distribution of
Okay, come in on
sclerites. Oh,
polyps.
I'm also seeing something really
Cover that
cool that
bridge.
we'll
Thank
look
you.
at after over on the right there.
Ship move in, pilots.
All
Understood.
right,
Thank
everybody.
you.
So what do we see? Is this a nodal brancher or an internodal brancher?
Looking for
I will
nodes.
give you all 20 seconds to answer. No,
So much
20
better
seconds
lighting
is
with
too
those
long.
lower
You've had
circle.
enough time. Internodal, very good.
Internodal.
Well done. And a whole cluster of barnacles just off to the left that are
It is so much
overgrowing
better lighting
on
with this
this
lower circloid.
colony.
Yeah.
And you can see the tissue just below those barnacles. Note that the tissue of the bamboo coral ends. And so the skeleton is exposed. And what that means is you now have a calcium carbonate surface for the barnacles to attach to, the barnacle larvae. And if you mind, can we zoom in? I'm interested in the branching thing just behind all those barnacles. I want to see if that's a sponge or a hydrozoan or a bryozoan. It's forming the backdrop there. Oh, it's a bryozoan. Beautiful. So notice the way so I'm looking at that finely branched thing behind the barnacles, and here you see there are zooids that are extending their tentacles, but you notice there's something wrong about the tentacles compared to all the octocorals and undercorals we see. They're a little bit rounded. Those are called lophophores. Same basic principle, but those tentacles are ciliated and they can generate their own current, and they generate a current swirling around them and they feed. Otherwise, it's a colonial animal. Boy, those ones are really hard to collect. I don't know. We don't have any bryozoan specialists, but I'll bet they would be interested in that. So that's a really good view. Thank you. We
Those are the ones that are really brittle.
Yeah, they are very
Yeah, they just
brittle.
kind of crumble apart.
Yeah, actually, bryozoans have a whole variety of colony form, and some of them are actually gelatinous.
Take it down.
So you'll find some, looks like a hunk of surf wax washed up on the beach. It's actually a bryozoan colony. So yeah, all kinds of different textures. So I was saying as we zoomed in there, just came in the screen far right.
Yeah.
That's a giant
Partial.
solitary
I'm gonna
hydroid.
get
Really
closer
cool. Corymorpha, I think. Corymorpheridae. Maybe somebody can remind me of the name.
Yep, so you can get your awareness.
Earlier, I think we did see, we saw that crinoid, and I said that there was what I call snail fur, this colonial hydroid that was overgrowing. This is now a solitary version of the same general group, the hydroids, but you'll see the polyps are giant. Oh, this is either 2 or 3 solitary ones next to each other, or they're all budding from the same. If they're all budding from the same, that changes the dynamics of what we call it. But still, it's a hydroid. So you see the main tube. It doesn't have a skeleton. That's one giant polyp. And then beautiful view here. The orange part is what I will call the hypostome. So it's a raised tube, and the mouth is in the center of it. And you see it's got subsidiary sort of feeding tentacles. And then it's got the very long tentacles around it that will be used to capture its prey. And then that fuzzy sort of flower bouquet that's ringing around the base of the hypostome, those are the gonads. And so that would be where the sperm or the egg are produced, depending on whether this is a male or a female, or possibly the species are hermaphroditic, I'm not sure. Fantastic imagery. These are always so photogenic. I think of them as sort of the deep sea radar dishes.
Do you have the extra lights on?
We do not.
Can we see what they look like with this?
Les
Sure
Watling
can.
is suggesting,
Colors now.
yeah, this Corymorpha is the name that I suggested earlier. I'm getting a couple of agreements. And Les says it's the first
Not
time he
sure
thinks he's
how
seen
much.
3 of them together like this. I do recall seeing 2 together.
Understood. Yeah, we got
And
the
it's
lowers
just an
deployed
interesting
again, so-
observation to make, and that's a nice cup coral off to its right. The number of them, because one of the things you want to think about is, well, how did they get here? And so they probably got here by a larva that was moving through the water column, settled on the surface, and grew. And so how did the second one get here? Did 2 larvae settle together? did the one that settled first produce a new larva, or did it asexually
that.
bite off another
Yeah,
one? And
actually,
these are
it's
the
off
sort of
and we're just
things
about
where
together.
we need to have
Same
observations
shot.
in order
That's
to,
good. There you go.
get to the bottom of that. Ideally, you want
Thanks,
to have
video.
some
That was awesome.
experimental data that
I
you
like
can
that one.
follow, but
Was nice.
that's in the future when we can
sunflower.
have more long-term deployments in the deep sea.
Okay, I think they wanted the single whip upper left.
Yeah, so we're going to the single whip just to now see, is the polyp structure any different from what we just saw on this one? Who knows what we'll see on the way, even though it's only like
So we
2
know
meters away.
about this one right here. Big guy.
I'm guessing that's like that one that we imaged that was just on the edge
Yep.
of the wall back
Okay.
there.
All right, I'll saddle over to the lone.
So this is certainly going to be another bamboo coral. And I can tell by how that skeleton is growing. You notice, unlike the primnoid whip, it's not nice and straight. It looks knobby because it's got these very large polyps, and so you're seeing some spacing in between them. And then as it's grown, it's kind of adjusting to the currents that are moving around it. And so the skeleton sort of looks kind of drunken as it's grown here. Rian Waller, our colleague, who's, I believe, still down in Chile, is tuning in. And she's an expert in some of the cup corals, and she said that we're looking at either Desmophyllum or Javania, that nice cup coral that was next to those solitary hydroids. So that was great. And she's just saying what an amazing coral rubble platform this is. And I agree, there's a lot of evidence of coral having grown here for a very long period of time.
I want to stow that part lower for me.
Understood. Port lower still.
Yeah, Rian says she's still in Chile. And Rian, I don't know if I said it while you were talking about, yeah, we had an identification of Selenosmilia here. It's a great view. I mean, the diversity and density here is such that I will have no voice left by the end of this dive if this continues.
Video come in. On spiral.
Yep, Ashton's going to have to
Hold
take
that.
over.
Who,
So
me? Couldn't be.
one thing I see is that, again, the density of polyps is really high. The tissue looks relatively thick. I get this feeling of, kind of rubbery
Oh,
appearance.
come out. I want to reset my toe. Thanks. 1 second.
Ashton, we have a question for you from the chat room from one of our Italian colleagues tuning in today. It says, speaking of rubble, is the dead rubble black due to manganese coating?
Yeah, I would imagine that part of it is definitely manganese coating. We're on about a 500,000-year-old seafloor, so I would imagine that we probably have potentially up to a millimeter of possible manganese incrustation.
I'll try to hold
It
that.
could also just
video.
be from partial lithification or remineralization.
It is max soon.
Understood.
So this is a terrific close-up we're getting, and once again, we're looking at the sclerites. In this particular case, there's no branching, so we don't need to
Try
ask
the
where
extra
is the
lights.
branching point arising from. And the needles in this one are shorter, and they're not
Extra lights will bring
dense
out the
in the
little needles
base.
in there, don't they?
Right in the center there, you can see one of the polyps on the upper side has 3 pink balls at its base, and those would be reproductive structures, either clusters of eggs or clusters of sperm. And what is going on? What am I seeing right here in front of me? Was there something on there? I can't tell if right in the center there's something on top of the polyps or my eyes are just playing tricks on me.
You can't tell if it's on top
So
of
I'm
the
not gonna
polyp
worry
or
about it
maybe it's
anymore.
a closed polyp.
It's almost like those needles kind of remain positive, proud of the, and then the polyp retracts and the needles are still proud.
Slight tilt
That
down
is
again.
absolutely what happens in some of them. You can see that actually kind of nicely
What'd you
on
say,
the
Ron?
upper right.
Slight
One of
tilt.
the polyps has withdrawn its
Yeah.
tentacles, and you can see that there are there should be 8 of those needle-like sclerites poking up. 8 because they would be in between each of the 8 tentacles.
Copy.
Great stuff. Les Watling says polyps thick at the base, few rods or needles, so probably base of polyps with lots of scales and collagen, which is why it's a little bit hard to see through. In some of the earlier ones we could very clearly see the pharynx. It's a little bit harder to see in these ones. So probably lots of collagen. Cool, great, good view.
Okay.
So
We'll continue
watch
on.
your upper on the part.
Full wide.
Okay.
Clear the fog lights.
Fog is clear.
Coming around.
Les was noting that in this one, we often see a little, it's almost like some of these polyps are wearing lipstick. Right around the mouth, we get this bright orange or red color. And this one, Les was noting that color
Switch.
extended onto the tentacles.
Of
and
course.
That's not something that he's seen
how much
before.
left?
It's on you. I think we go to 1400.
Not sure.
So
Not sure
70
what that
minutes.
was paddling through the water column.
Perfect. All right.
Just to the right.
Sure.
Far left.
After this.
Probably our first Iridogorgia of the cruise, I think. So this would be another member of the family Chrysogorgiidae, but they grow in these beautiful spiral shapes. Someone will have to remind me if we've seen the Iridogorgia already. But this is often one of the favorites of folks who are watching the deep sea dives because they just have this really elegant appearance to them. In my opinion, all of the Chrysogorgiidae have very interesting geometry associated with them. So in the genus Chrysogorgia, the main axis turns at these very specific angles, but very angular, where this one turns in a very smooth spiral. And so you're looking right down that spiral. How cool is that? And coming off of the spiral, just on one side, are these branches, and the polyps are aligned on the branches. Sometimes we see the polyp on the main stem.
Understood.
I'm not seeing that on this one.
Got the extra lights on?
Yep.
Fog's coming on.
Quite often there is a shrimp that's associated with Iridogorgia.
Take one. You got it?
Yeah, if you zoom, go towards the left. I've seen something on one of the branches. It's kind of a hot pink in
Try to say
there.
that. Later on.
So left.
Yeah, it's also flexing left too. So
It's probably an amphipod. But one of the things, a hallmark you see about the Iridogorgia is that they have these very thin skeletons, extremely thin layer of tissue on the skeleton. So you're basically seeing the skeleton. And then the polyps are almost like little teardrops.
Yeah.
Oh, there we go.
See that.
That's a polychaete worm.
Trade off.
It looks like a scale worm in the background there. I'm looking at the second layer of branches. Nice.
It's flexing in the wind, and I'm trying to track it.
Yeah, no,
Come out video.
that's good. Thank you.
All right.
Can we have the lasers on?
Yeah.
Briefly. So one of the ways you can tell apart some of the species is the diameter of the spiral. If it's a really
Okay, Chris,
tight coil or
get
if it's a
ready to
really
take it.
large
Understood.
coil. So for example, one of the common
Do a
species
quick
here in the
trade-off.
Atlantic is Iridogorgia magnispiralis, which means wide spiral. This one did not have a wide spiral. My initial guess would be Iridogorgia splendens, but I think Les Watling knows a lot more about these than I do. Yeah, and he's just put in the chat room, probably Iridogorgia splendens. Yeah, if you were watching on the very first dive
So you're pretty neutral here.
of this
Yeah.
voyage to the Ridge
We get 5
expedition,
meters to
we saw a whole
5.
lot of yellow black corals that formed a very tight corkscrew spiral also. They didn't branch. And those black
Bridge, are we
corals,
now?
all the
You done,
polyps were
Fox?
on the inside of the coral.
Probably.
Where here, all
We'd
the
like to
polyps
put
are on
another
the
ship in.
outside of the coral on those extended branches. Kind of
Range
interesting.
5 meters at a bearing.
Different
285
strategies
degrees,
for
going decimal
Picking
2
food
knots
out of the
always.
water column and interacting with the way water flows over you.
So if you don't have bottom lock,
Thank you.
Yep.
then there'll be red text, and the auto X will be grayed out. So I'll give you feedback whether or not you're go or no go.
Copy. Thank you.
Let me bring your port lower back upward.
Yeah, it looks like it's mellowed out a little bit on the terrain.
Upper swing arms, lower swing arms. Here we go. Deploying.
And what we're seeing is more of this
Lower.
black coral skeleton.
Get that
So
light
the skeleton
spread
of
out
coral
nicely.
that's possibly
Okay.
likely encrusted with a manganese coating or partially
Typically, we leave Valpex
remineralized,
disabled.
giving it
Understood.
that dark black color. And it's kind of just in these low areas with sediment underneath it. It's biogenic sediment that's produced by fecal decay or fecal transport from the upper water
That
column.
looks good.
And then around those, on the kind of higher portions, we have these angular fragments
5
of
meters coming
basaltic
towards you.
pillows and this is pretty
Copy.
standard
On
that
the
we
way out.
see in these highly faulted
30 meters
areas
from the
on
slip.
the
So great.
upper portions
Looks like it's,
of
like you said,
these
leveling off.
foot walls
It
of these normal
measured it.
faults. As this
High-pack agrees.
portion of the oceanic crust is faulted, and this is moves upwards relative to the portion that's more ridge center. There's a huge amount of shearing force in that. Think about just
I gotta work
breaking
on my
all that
floating
rock.
shots.
That's pretty good.
I know. It's fun.
Go ahead, Ridge. Thank you. Move complete, pilots.
Scanning with blue view. Thank you, Nav.
Lower right, you see the round?
Coming into center.
No, below that, directly below, near the bottom,
Understood.
like a husk,
Guide me.
right next to the edge. of the
Pilots,
bottom.
if you can you zoom
Yeah, between
in
lasers.
upwards? There's a
Now left corner.
if you keep going up.
Understood science.
Oh.
Video was looking at something here.
Sorry. Yep. You see that kind of angular piece of crust that's sticking up? It's on the same horizontal line as that sponge in the middle, but it's on the right side of the lasers. The lasers are kind of just to the left of it.
Understood. On lasers now.
It's right below the lasers. Yeah, so
Just below
that
the
and just to the left. There's kind of that angular piece, and then there's a little smaller piece to the left of that big one. Yeah, that. Can you zoom in on that for a second, please?
Take that video.
I want to see if it might be grabbable,
Partial.
or anything in there is grabbable. It would be nice to try and grab one of those pieces of
It's Nav.
fragments.
Understood. Slipping back. Stand by. Bring partial video.
Full right.
Come up to it a little bit.
Pretty calm out there.
Yeah,
13
so
knots
those
on the bow.
pieces that the laser were on are they look like the outer kind of skin or thicker portion of these pillows. So maybe where a pillow formed and then lava inside of it drained back or continued
video.
to flow,
Come
and
in.
those surfaces tend to be glass rich and good for sampling
Partial.
for further analysis.
So you're thinking science just a little to the right here?
Yep, a little to the right. I'm kind of looking for those thicker crust areas. That dark one, I'm guessing, is probably pretty big.
The one dead center?
Yeah, the one at your 12 o'clock to the lasers
Understood.
with a little shadow underneath it.
Yeah.
Chris, have you sampled a rock before? Okay.
That one on your 3 o'clock that's really dark surface is nice, but I'm guessing it's pretty big, looking at the scale of the lasers. So
Yeah, maybe that little guy.
yeah,
I could pan up to just to the right of lasers here. Little circular one. I don't know.
They typically like sharp ones.
Understood. Yeah, I don't see any good specimens in here, science.
yeah. What I'm looking for is usually think about something that's almost got the shape of the rind of an orange, right? It's that kind of outer portion. That's why I was looking at the one with that little shadow next to that dark one.
Understood.
But I imagine a lot of these you kind of just have to go up and nudge and see if they're attached or not.
Very partial wide video.
Partial wide.
All right, science, I'll let you tell me if there's something that you want me to poke at. See
Yeah.
if it's loose.
Can you poke at the one at about your 4 o'clock?
4 o'clock. Just bottom of the screen.
Yeah. Stop there and then go horizontal to your right.
Understood.
Keep going. Keep going. Keep going. Yeah, that guy.
Right there at dead
Yeah.
center.
Yep.
Lasers on. Stand by. Just settle out.
So, I see the arm down here.
Yep.
And I see your lasers way out there.
Too far.
So if you want to poke that rock,
Come in.
you're way too far away, I
Yep.
think. You agree?
Yeah, I think get full wide on video.
You're full wide.
Okay, yeah, it's a little far out there.
I think it's deceiving. We could. Am I zoomed in on Mini Zeus at all? No? Okay.
Negative.
All right. You come back up and get a little closer.
Yeah, I'd hop a little closer.
Understood.
Maybe put your toe like where that ball is. Right?
Yeah.
Or like even a little below, maybe a little below that.
Yep.
Yeah, I'll drive the camera here. There you go. So you can see both the toe and the subject.
Yep.
There you go.
Somewhere there.
Yeah. Jocks are up, 1,000 RPM.
Understood.
Retracting drawer. Okay. And then you can ask them. Sometimes they want a squeeze test, which means like squeeze it with a high grip force and see if it crushes, and then they won't want it.
Yeah.
But we'll see what he asks for.
I'm creeping a little bit, just trying to see if that settles in.
Yeah, just listen in. I squeeze test won't be necessary. It'll
Understood.
be more of a can you rip it off test, or is
Is it stuck or not?
yeah.
Okay. I think we're settled out there.
Go ahead, Bridge.
And then you're familiar with the jaws closed
Yep.
loose test? Okay.
Sure am.
All right.
Okay. Hydraulics coming on
Copy
now.
that. This time, is there anything we should do down here?
Hydraulics on.
Copy that. Thank you.
All right, you might have to guide me in. I think I've lost sight of where we're going here.
It's basically right behind the jaws there. In there.
Directly
I would
behind.
just start, scratching once he says. Any ones that look loose, scratch
Give me
it.
a little partial video
Ask
on
him
the
to-
main. So you're looking in here, Ashton.
To your right, off screen
Come back
of the claw.
Yeah.
Yep, the one that's just to the right of that. Yeah, you're almost on top of it now.
Yeah, that's
Yep.
that guy.
Yeah, right there.
That's the one we want.
Let's grab that.
Understood.
Do you want to do port box and open it?
Yeah.
Okay. Port wing coming inboard for you. Oh. Okay. Let's see, I can inboard a little bit. Let's see how it started hitting the.
Yeah, that's good there.
Can you get it from there?
I think so.
It hit that rock sitting there. See that?
Sure did.
Nice. Okay, following you back up. Vinicius. Thank you. See. Vinicius will have a pretty good angle today. Looks like you're in grip force 5. Should be enough.
Let that dust settle just a hair.
Okay.
Coming just a little video.
See the jaw hanging on that one in the background. Oh, okay. Slip down. There you go. Nice. Now just go straight to the box.
Yep.
Okay.
Do we want to image it first or just put it away?
That's up to you, Ron.
Actually, it's science. They use it to identify.
And let's get a photo of it actually real quick, just in case.
Understood.
So keep an eye on your
Anything else
elbow
in there?
relative to Vinicius.
Yep.
Okay.
Let's kick on the extra lights.
Okay, coming up.
Get a focus.
So yeah, just be careful here.
Right, yep.
Good, nice slow rotate. Maybe a little bit faster. That's good.
Give all that biology a chance to escape from it.
Yeah, I was going to say, it looks like there's something wiggly on there. Oh, and not anymore. That's the interior of the rock. Well, kind of looks...
I'd put it in the box.
Is that the
before
interior, or
drops.
is that on the outside?
That's
Oh, that's
the white.
on the outside.
Yeah,
Okay,
yeah.
okay.
But it looks nice from the standpoint of it looks like that maybe that exterior rind of that shell, of that pillow. So, you know, if we're going to have a chance to see anything kind of glassy
It's pretty good too.
and not very crystallized, that'll be an opportunity. Especially, you know, hoping it came from that really large
Nice.
cross-section pillow that you see in the foreground.
Sure.
Thank you, ROV.
What's the brown stuff on the top of this rock here? Is that just some
Wanna
other
close it?
mineralization?
Yeah, let me close it.
It doesn't look like
I think I can close it.
mineralization. It's, you know, I think we talked about it the other day in terms of how these rocks are exposed, both in terms of whether they've been partially buried or whether they're freshly fragmented, can affect the precipitation on them and the length of time they've precipitated out.
Understood.
I don't know why I'm still having such a hard time remembering that
Nice.
all this stuff wasn't just formed at the surface. You know, and
Yeah.
that's still kind of in my brain. I see it there. Well, that's probably where it was
Take-
made, and not that it's been uplifted or transformed. And
Yep.
I have to keep reminding myself of that.
So-
Or listening to you,
So,
Rodney.
so it, you
Take
know,
a
it
run
was
on that?
all formed at the surface of the ocean, but then covered in more
Rocks
things
disciple.
that were, you know, erupted
Disciple.
on the surface of the ocean.
That's a good point. Right.
So, you know, if you think about the oceanic crust, ignoring the sediments on the top of it, because I like igneous processes and not necessarily sedimentary processes, so we'll just throw this out the door.
Sorry, just hang on a second. Pilot, before you go too far, just right of center, there's that thin stick crinoid again, but this time with nothing growing on the stalks. I just want to see what the stalk looks like so we can compare to earlier. Thank you.
Understood.
Go ahead.
Yeah, so you can think about in this area or
I turned them
throughout the oceanic crust, the top kilometer to two of that oceanic
wanted
crust
them
is
off,
formed
so
by
I'm
these
sorry.
pillow
Well, I
basalts.
did.
So
I turned them off and then-
just kind of pillow eruption on top of pillow eruption on top of pillow eruption. So they're all formed at the surface, but then
Take
later
partial.
buried.
Right.
And we haven't really seen any exposures of what's below those, which are more what we call dike features. So those are the feeding systems between the oceanic magma reservoirs that are feeding those pillow basalts, and those will look very different. They'll look, you get a lot of columnar basalting and formation.
High pack. What's the D2 scale normally set to? Do you know?
Interesting. This crinoid again has all kinds of stuff growing on it. It's got those hydroids at the top, and it looks like
D6. Let me
I'm
check
not sure
real
if
quick.
that's
Okay.
the bryozoan
Check one
again
video.
down below. And then this looks like maybe an egg string or something. Yeah, interesting. And you can again see the tube feet sticking off those side branches there. You can see the tube feet really nicely. They're the lightest of the
Copy.
I don't know, call them branchlets on the arm.
That looks
Now let's scan down towards the base of this. I want to look at that branching thing that's on there. So is that the... Yeah, no, you just passed the branching.
Understood.
There we go. Yeah.
Max zoom.
Well, if it is the bryozoan again, it's not nearly in as good condition. I don't see any of the zooids out. Okay, thank you, pilot. We can move on.
Understood. And everybody is clear for one.
nice. You want to give me 5 meters, 285? If you're good with it, pilot?
Yeah.
All right.
5 meters, 285.
Copy that, will do.
And we have the Bathypathes black coral that was just on the left of that
Bridge,
crinoid
okay.
that we imaged. That crinoid is so interesting. I'm trying to see nicely its stalk in
Range
case there's
5 meters.
taxonomic characteristics
bearing
associated with it,
285
but both
degrees.
times that we've zoomed in,
Decimal 2 knots,
it's been
please.
completely overgrown by stuff, but not the upper feeding part of the colony. Excuse me, it's not a colony, that's an individual.
Ship move is in.
Copy.
So, looking out on the landscape here, there's lots of coral rubble. Much of it is blackened, and we talked about this manganese crusting. And then you see these occasional yellowish and light brownish upright colonies. So that's the live stony coral. Selenosmilia is the name I'm getting. Sort of pinky orange, highly branching colonies. Those are bamboo corals. There's one smaller one just passing the center now to the left, and there's a much bigger one coming in on the right.
Oh, you found a good little nook here.
There's a whole series of individual whips.
See it. And
the orange ones are Aphanosticopathes.
It's hidden around the corner.
You can see several of them just up top left of center. Lots of white sponges. But I'm seeing a lot of these Selenosmilia colonies, and you know that's where all the rubble comes from. So again, just suggests to me that they've been in place for a very long period of time. The kind of creamy white fan that's up at about 11 o'clock, it's growing off just below that white sponge. I think that's the Anthothela again. And then on top of the sponge, the brighter yellow one, that's probably the Acanthogorgia. See pockets of the bright purple overgrowing stoloniferous form.
Go ahead, Bridge. Thank you.
Interesting suggestion on that previous crinoid we're looking at. There were the hydrozoans at the top, that sort of snail fur, the Hydroctinia, but not lower down. And Tina's suggesting that maybe somebody was grazing on those. There's another Iridogorgia colony right in the center. The whip upper left is our primnoid whip, possibly convexella. Can we have a look, upper left corner, that white sponge? Looks like a vase kind of tipped over, or like the amphitheater sponge that we collected yesterday. And just like yesterday, we see that there's one of these stony corals growing on the inside of it. And, you know, I still don't really know. We have this question about which one is overgrowing the other. This one's kind of interesting. We'll get a nice close-up view for our sponge biologists to have a look at later.
Understood. 5 meter move
So,
should help us out.
A little bit of a tough area to get into. We don't want to damage any of these corals.
Oh, I was talking to Pilot.
So pilots come above this and we'll zoom on it going down.
Answer's clear.
Understood. I can turn them off for you if you want. Lasers are off.
I'm not sure if this colony on the right, this must be another plexaurid. So what I call multiflabellate, which means there are multiple fans, one behind the other, so it kind of looks like a bush, but it's actually a planar growth form. All right, let's look at the texture of this sponge. Oh, you can see lots of these openings. And I think the openings we see are all X-current openings, not in-current, but might be a little bit of both. Usually the in-current openings are smaller than the X-current ones. And just remember that it's got cells on the inside that have these little whip-like flagella that constantly beat and draw in a current.
You're filling the frame on your wide-angle cams nicely.
There's a very
It's awesome view.
sort of typical Paramaricia fan on the lower left.
There you go. And you can look inside the dome of it.
Yeah. And let that go.
It's probably an anemone overgrowing the tip of that Lophelia, just down in the center there now.
Yeah, I saw it, felt like that too. A little bit of current or something.
Yeah.
Seeing lots more sticks on the bottom, several of which are crinoids, but then the whiter ones, the lighter color, those are the primnoid whips. Another one of these interesting sponges. I'm not sure if these are conolasma. I would have to look that up, and right now our sponge biologist is in another meeting.
And kind of follow the good stuff.
Yeah, if you want to come around just a little bit, I think everything's over here. Thank you.
I feel that way too.
I'm seeing just right of center another one of the anemones. I thought that's what I saw on the Selenosmilia we just passed, and now I can see it head on.
Take partial there,
And
Rob.
it's kind of
We
closed
get
up,
5
so its
meters
tentacles are
bearing
withdrawn.
315
So when you look at a
degrees.
sea anemone like this one, a sea anemone is simply a very large polyp that doesn't have a skeleton, that lives on its own. But it's otherwise similar to all of these stony corals and octocorals in that it is a cnidarian, it has soft tissue, it has stinging cells, it has a single opening into the body cavity, which is a mouth, and that is ringed by tentacles that it uses to capture its prey.
Just creeping in on it.
So this is a fairly large one, and I wonder if it's captured something recently, and that's why it's closed up like that. Not sure what.
Copy.
Small crustacean, a small fish, worm,
There,
something.
Rowan, you can take a little more.
It's kind of funny. He's got
Yeah,
little
really nice. It almost looks like it's biting its
failed
own
to
tentacles.
retract with its tentacles.
It's kind of weird. But it looks quite bulbous. So, best guess, it's got something that's digesting. That really nice white colony, branching colony to its right, that's another of the bryozoans. And when you get a good close look at bryozoans, one of the things that's cool is each of those zooids is in its own little case or house. And so you can almost see that there's a series of boxes that make up the branches, and each of the zooids is coming out of that. Thank you. And when video is finished, as you pull out, I would like to look below the things that we're seeing here. I noticed something else coming in, a bright yellow right there. That bright yellow sort of fanny stuff. I'm not sure if that's a black coral or hydrozoan, but it's certainly something we haven't seen today.
Maxim.
Understood.
I am going to say bryozoan. I can see on the tips of some of them, they seem to have those arcing, curved lophophores. Yeah, you can really see it well on the left side, pointing down. So this is at least our second, maybe our third kind of bryozoan colony that we've seen this morning. So fantastic video. Bryozoans are really small. In fact, as we pull out, we'll put the lasers on, but don't pull out quite yet. Just look to the lower left off the screen. Now there's another kind of octocoral that grows in tubes, and the polyps stick out of the tube. Just go to the left just a little bit. There we go. And now you can see them elongated. They're sticking out of tubes that are, I believe they're chitinous. Chitin being the same molecule that forms the exoskeleton of crustaceans. Excellent stuff. Yeah, so when we zoom out, if we can get the lasers on the yellow, just to get a sense of how small these are and why it's so difficult to see them. But when we have, you know, this great cameras with the ROV and the lighting, we're able to get a good sense. So that's maybe the whole colony is perhaps 10 centimeters across, about 5 inches. Really delicate, as you saw, and it was made up of, you know, several hundred at least polyps. Thank you. Let's move on.
Whenever you're ready.
I'm seeing another paragorgia down in the corner, the bubblegum coral. Oh, this might be a chimera in the background, that fish. That's probably the first chimera we've seen of the cruise.
Take that roll.
Those are good ones.
Yeah, absolutely. I know them as rabbit fish. These are related to sharks and so on.
There you go.
Very nice. They're very slow moving. Very cool. You see the lines that are dividing the head. Those are lateral lines. They're sensory pores or sensory cells in there so they can detect vibration and movement in the water. I'm not sure if any of them can also detect molecules, which would basically be a sense of smell. So while we're getting this great video, let's see if I can pull up this note that Ken Sulek sent me earlier today. Yeah, so he was saying this information with respect to a different fish, but I think it applies here because of the way that the eye is. He says, These eyes are characterized by a phakic space, meaning the dark retina is much larger in size than the lens in front of it. And that's a way of being adapted to low light condition. It has two known functions. It increases the surface area of reception of photons, and it enhances binocular vision, and especially if it's got this teardrop shape.
Bridge, are we now?
There, going. Take a look, going.
Would like to put another ship move, please. Range 5 meters, bearing 315 degrees at decimal 2 knots, please.
Mike Vecchione is helping me here. So these various pits and pores that you're seeing, just like sharks, they have electroreceptors on the head.
Copy.
So they can also detect changes in conductivity, I guess, called the ampullae of Lorenzini. It's also nicely passing over a different kind of black coral there behind it. Yellow sea star underneath it.
Video's clear.
Very nice. Mike Vecchione says that those electroreceptors are good for near-field detection of prey.
Those are cool fish.
Yeah.
So we'll look forward to Ken Sulak's report later on that fish.
This is always helpful.
Yeah. I put one more little 5 meter move in.
Yeah.
Several yellow colonies here. I think that yellow colony, yes, oh, absolutely. So let's, do you see the white sponge just above the lasers? To the right of it, there's something bright yellow.
Oh, the tether you got.
And that's going to be a parasitic zoanthid overgrowing a bubblegum coral. And that's what it's also going on in the lower right corner. So that's our first observation of this today. So this will be an example to show you that within the cnidarians you can have various lifestyles, and one of those is basically parasitic, where one cnidarian is taking advantage of and usurping the energy of another one.
Understood. All right, Science, we're just waiting for a little bit of tether here. We'll
Copy that.
get a little closer.
No problem. We're just waiting for some tether, which means that the ship has to move so that Sirius can move, so that the cable that D2 is on can stretch far enough for us to get the good views.
So this is what we're looking at here.
Yeah, I was talking about the one at the top left, but this is the same thing right in the center, so it's closer. Let's go for it.
Understood.
Thank you.
All right, roll. Take partial.
See the lasers on there, 10 centimeters apart. So this is a bubblegum coral. That's the underlying red that you see. The swollen areas are where the polyps would be. All the polyps are currently retracted. And then the yellow polyps that you see growing on top of it are zoanthids. So those are the ones I was referring to as parasitic. I'm saying parasitic because sometimes we see a colony is completely overgrown, and so it basically kills that colony underneath. Here the Paragorgia is still alive. And there are some zoanthids that are specialized for growing on sponges, some on corals. I think the one that we're looking at here is a specialist for coral. I don't know what species this is, but there have been some identified, for example, in the Pacific. You can also see on these zoanthid polyps, the body wall looks very rough and bumpy. And that's one of the ways, actually, that I can tell that it's a zoanthid and not a sea anemone, because otherwise it looks an awful lot like a sea anemone. Another difference to the sea anemones is that you see that all these zooids are connected to one another, so they are truly colonial. They're asexually reproducing. And then the other thing we see here are these snake stars or basket stars. These are brittle stars, ophiuroids, that are specialized to live on these octocorals.
Center up for full screen.
Understood.
Great, thank you. Good view. Les Watling believes that this is the species Paragorgia coralloides. And we have seen this relationship very frequently here in the North Atlantic. The work that we've done on the New England seamounts, which is a chain of seamounts that kind of extends from the northwest to the southeast, moving from off of the northeast coast of the United States and moving towards the Mid-Atlantic Ridge. And we see this quite frequently. Yep, there's a white sea star. I saw a bright yellow one a short time ago. There's an interesting question there about that relationship between those two. Is how fast is the zoanthid going relative to how fast is the paragorgia growing, and that'll determine if the zoanthid can eventually completely overgrow the paragorgia. I do know that there are some zoanthids that can overgrow their coral host and then put out their own skeleton, but most of them don't do that. So this fish, is this a Bethydid? For some reason that name is jumping into my head. Very rounded head, snout, bulbous head. Okay, thank you. This is interesting. Les Watling is saying that this observation of that zoanthid with the paragorgia would be the furthest east record known for this interaction. But going westward, they extend all the way into the western central Pacific. So very cool. If you want to look at something new, I'm not sure where you're going. Oh, you're going after the fish. Let's look far right, just the edge of the screen right now. There's a large crab on this fan coral, on the front face of it. Boy, as I look up the slope here, I feel like I'm in Alaska in the Aleutians, where in the Aleutians we have a very, very high density of coral and sponges, which is reflective of the very high productivity overlying the water. I'm not sure that me personally in the Atlantic I've ever seen this much coral in one place. It's incredible. Yeah, so this is oh, I said it was a crab. I'm sorry. This is another kind of brittle star. One that kind of fishes in the water column. You see that it's got its long arms extended there and they're coiled. And so I think what it's doing is putting its arms out to try to snag things, zooplankton, that are in the water column. I don't think that's an astroschema, but I'll let my colleagues on shore correct me if I'm wrong. The one to its right, you see it's got its arms all wrapped around. That one's definitely astroschema. And then here's yet another kind of brittle star. You see how spiny the arms are, how different that is from the one to its immediate left. And then it's wrapped around a crinoid. Both of those are types of echinoderms, but they're in different classes. The crinoid is a suspension feeder, has its mouth facing upwards, and always uses its arm to grab things out of the water column. The brittle stars in the Ophiuroidea have lots of different ways of feeding, and it depends on the species. And so I expect the one with the spiny arms feeds on zooplankton or on this colony. The one that has its arms extended is fishing, and the basket star is an associate or of this coral, always spends its life there and probably feeds on the mucus and whatever's getting trapped in the mucus. Meanwhile, the coral itself is a plexaurid, maybe Paramercia, I'm not sure. Yeah, so 3 different species of brittle stars all together there on that same coral. If you're in a stable position, maybe we can zoom just above this fan that we just looked at, and there's something kind of teardrop little polyps, delicate polyps, 2 bottle brushes just behind it, right in the center. This is going to be another kind of chrysogorgia, and what I'm trying to see is that 2 colonies or is that 1 branching? Yep, you're zooming right in on it. This pink thing. This looks like something else that we've seen on the New England seamounts. So I'm seeing a lot of things that remind me of the New England seamounts mixed in with some new things. And one of the things you can see on some of the polyps here, notice how swollen they are at the base. Yeah, just coming into view at the top, they're quite swollen. And that's an indication that they're going into a reproductive state. I might say something about the overlying productivity in the water column. There's lots of food that's being produced, the polyps are feeding on it, and they're diverting a lot of that energy into making eggs and sperm. And so the polyps get swollen. Basically, the gametes are stored inside the digestive space on sheets of tissue that subdivide that space, and then when the appropriate time, they'll release the sperm and eggs into the water column for fertilization. All right, so maybe Les Watling will recognize this. He's described some species of Chrysogorgia. Actually, the one on the right, the one on the left, you can see this, what I'm talking about, this angular spiraling to the branch. And one of the characteristics that's used, I was trying to describe this to Ashton the other day, if you think about that main axis and the branches coming off of it, that main axis is kind of twisting, and it twists at a very specific angle, and every once in a while it puts off a branch. And they sort of measure how many spirals does it take for the next branch to be directly above one below it. And so in this case, I'm going to guess that it's 3 branches for it to come out, and which way is it spiraling, right or left? So they would call it a 3 left something. Okay. No rocks you want to talk about here, huh?
A 3 left what?
Yeah, exactly. Something, I believe, was the technical term I used.
Do you have similar, I guess, nomenclature that they do in chemistry for enantiomers, R and S, depending on the rotation?
Right.
Right-handed, left-handed?
Sort of thing.
Yeah.
Yep.
See, I'm paying attention.
That's good.
Yeah.
Somebody is.
I'm trying to take this opportunity to advocate for all the next dives that we're going to do in all these amazing geological places. Yeah.
Oh, let's look at the white fan just to the right of center. Just, it's waving at you right now. It's behind this stick. That looks like something new. I'm going to predict in advance that it is another Primnoidae. Asako is asking the same question. Is this a Primnoid fan? Yes, absolutely. This is in the family Primnoidae. I'm going to guess that this is the genus Candidella. And if it is, one of the things that we typically see on Candidella are polychaete worm associates. And I'm not seeing them here. Usually when they're present, they induce the coral to lay down some sclerites in the forms of little tunnels or garages. And I don't see that on here. But you can see with this lighting, there almost looks like there are rings going around some of these polyps, and those are scales. So I'll reference again this word sclerite. It's a microscopic calcium carbonate secretion of the coral, and they come in just in a wide array of shapes. In this family, the Primnoidae, they deposit those kind of like a suit of armor. Think of a medieval knight. And so the rings you're seeing are those different scales, and we can count the scales, look at the shape of the scales, ask how many rows there are. All of those will be keys in identifying the coral, how it branches, and then do the polyps come off singly, or in this case they look like they're whorls of 3. That is, there are 3 polyps wrapped around a branch. Great, thank you, good view. That shrimp, Les identified the one earlier as Bathy paleomanella. A lot of these deep sea animals have the word bathy in their name. You've heard me say bathypathes, for example. We talked about a Ctenophore bathysiroa. So if you heard the very beginning of the dive, our pre-dive briefing, I said that we're in the bathyal zone. So this bathy or bathyal refers to a zone of the ocean, a depth zone of the ocean from approximately, I think right now the thinking is about 800 meters down to about 3,000 meters. And we have these zones subdivided, and they tell us where we find major changes in the communities of organisms that live there. Pilot, if possible, zoom on the branching brown coral right now, dead center. Thank you. I have one of the scientists on shore asking me about a black coral, but I'm not sure which one because there's black corals all around us. So we're going to start with this one. Anyway, so a lot of the organisms are named after that depth zone, bathy, with some other element to the name to distinguish them. So I think we were looking at something like this earlier. I referred to it as a branching parentipathes, and it had, more than a dozen chirostylid squat lobsters on it. I don't see any squat lobsters on this one, so I wonder if this is something different. I wonder if this is something like taxipathes or whatever. And we're really lucky to have Tina Molosova watching, and she's transmitting some information, and she can give us an ID on this. Interestingly enough, this also has a brittle star in its branches. No squat lobsters. To me, the polyps here seem thicker. I don't think that this is parentipathes. In fact, I'm quite certain it is not. So I'm looking forward to the name Trisopathes. Thank you, Asako. Great. So that's the first of those that we've seen today. Excellent. Pilot, as we continue to move here, have a look over to your left. You can zoom out some more. This is something I want to point out that I think we'll all be happy to see. Just at the base of this sponge and on the other side of the sponge, notice there are two of these branching octocorals that have yellow stems with blue polyps. So I see three right in this area. So I wanted to highlight to everybody that we did not sample the only version of that coral on this seamount. Now, we don't have to look at it. I'm just referring to the fact that there's lots of them here. I'm really pleased to see that, because before we saw only one and we sampled it. It's good to know that it's actually several of them. In fact, there's 5 or 6 of them right here. That's fantastic. Here's another bubblegum coral. This one not overgrown entirely by the zoanthid, but there is some of it, and there's several brittle stars still on it. So here you'll see an early stage, I think, of this overgrowth, or maybe there's none. So here's an interesting question. We've often said that the presence of these brittle stars on these corals, the ones that are seem to be in an associate-host relationship, that they feed on and protect and clean off the coral. So why aren't these brittle stars presenting the zoanthids from overgrowing this coral? Why aren't they getting the initial larvae that settle and start growing? They're not doing their job. Rian Waller says there must be some serious productivity in this area. I agree, Rian. 3 dives ago, you weren't able to be with us, but we dove on a deep axial volcano, and we were stunned to see that it was covered in pteropod ooze, and we expected it to be really fresh basalt, and it's just an indication of how much overlying productivity there is in the water. And the only way you can have all of this coral and all of the sponge blanketing the surface of the rock, of the seafloor, is if there's all kinds of food being produced above it in the water. Because the source of the food is the sun, the phytoplankton that are using the sun's energy, and then they sink down here and are fed on by this. This is unbelievable.
I was reading that the average time to make it to the at least abyssal depths of the seafloor from the kind of upper photic zone is about 2 weeks for a fecal pellet. I don't know if that's...
Is that right?
Yeah,
yeah.
Revised, that's right.
So there's a lot of interesting study on what's producing the fecal pellets. Mm-hmm. So we're talking about basically zooplankton like copepods and the salps and the jellyfish. They're all feeding up in the surface of the water, and then they poop out their undigested remains. Well, that fecal pellet that they've produced has a lot of carbon in it, and it sinks faster than the phytoplankton sink on their own. And depending on, not to get too gross about it, but how firm that poop is, it can sink faster or slower. And so there's a tremendous amount of study on who is doing the feeding and what is their fecal pellet, because that translates to how fast the carbon is being removed from the surface of the water to the depth. And one of the things they're seeing is that with climate change, we're changing the communities of zooplankton in the upper water column, and so we're changing the rate at which carbon is sinking in the water column. It's really fascinating. But back to this magnificent view, these are the beneficiaries of all that food sinking through the water column. Rian Waller's asking if it's been all Selenosmilia or if we've seen any Lophilia. All I can say for certain, Rian, is that I feel like I'm seeing at least 2 different color morphs among these branching stony corals, and I was interpreting that as Lophilia versus Selenosmilia, but I'm not certain. And it may be that we saw a few colonies of Madrepora earlier, and now we're seeing Selenosmilia. I'm just not confident enough in my ability to separate them. Lots of these primnoid whips that we're passing over, a few of the black coral whips. They're the ones that are coiled, irregularly coiled. I see an orange one down there. An Acanella bamboo coral. Some beautiful vase sponges. Some Euplectella, the tube sponges. Our depth right now is 1,724 meters, and when we first settled down on the bottom, we were at 1,759, so we really haven't gone up very much on the slope, and that's because we've been so overwhelmed with the diversity. There's so much to zoom in on, we're not making much headway. And I apologize to my scientist colleagues in the chat room if I miss your chat. I'm trying to go back and forth, but sometimes, especially when there's a lot of observations in the chat room, I may miss it. If Carlos, Tomo, Marina, Manuela, if any of you are in the ECC and would like to share any observations of what you're seeing, please feel free to speak up. We're zooming in here on an asteroid C-star, and it looks to me like it's in a feeding position. You notice the way it's sort of bunched up, and I notice on the right side of it, it looks like I'm seeing some stripes. So I wonder if it's pushing down a little coral on the rock. Actually, those are probably its tube feet that are extending from its legs. So as we look between those arms, the leftmost arm, I'm seeing some white globules kind of below it. So I wonder if that's what it's feeding on, if it's pushing it down, so that the arm that's like pointed at us is along the branch of whatever that is, or the axis of whatever that is. Yeah, I can't quite tell from this angle, but it's definitely on something. It's absolutely feeding on something. And I believe it's a coral, so I see one polyp there that I think that's a polyp of an octocoral, so I think I see sclerites in it. I'm hesitating slightly, want to leave out the possibility that it's a sponge. Suggestion, the sea star is in the genus Evaplasoma, which is a very common coral predator here in the North Atlantic. You can see the tube feet underneath that it uses to move around and to grasp its prey. The red dot on the end of the arm that's pointing at us is sensory. There's pigments in there so it can detect light. And then as you go back along the arm toward the upper surface, you see there's a smooth dot in the center. That's called the madreporite, and it's basically a sieve plate that allows it to exchange water with the seawater into the system that's controlling those tube feet. It's all hydraulically driven. Pilots would probably appreciate, excuse me, that all these echinoderms use a lot of hydraulics for moving and manipulating. All right, that's good with us. never tell the time on that clock.
Now speaking of, I guess, hydraulics and the ROV, and the ROV is under an immense amount of pressure. We're at a depth of about 1,700 meters, so it's at a pressure equivalent to about 170 atmospheres.
Sorry, hang on. Before we leave, far left, there's a rounded, I think, sponge, lower left corner, just, by that purple thing. That might be a sponge that we have not yet seen this week. So, sorry, Ashton.
No, so I was just going to say, the ROV electronic system is, a lot of it's encased in oil because it allows it to maintain pressure from the outside water static, the lithostatic pressure of the water, but also is not conductive. So are any of these organisms analogous to that? Are any of them filled with like a mucus or anything that prevents, for lack of a better word, conductivity or any type of electrical signaling, but then maintains their structure?
So I'll get back to you on that in just a second. Pilot, I just want to inform you before you go on the sponge, the top of the screen, there's a sea spider walking down towards us. So we'll get back to that question in just a moment. This is Colossendidae is the family for these sea spiders. Pycnogonidae is the larger group that they're housed in. They're always so photogenic. They're fascinating animals, most closely related among the arthropods to scorpions and spiders and other arachnids, but they're not directly in that group. And what you see in the front there, that very long red tube, is a proboscis. And these are actually suction feeders. And you'll stick that proboscis into a coral polyp or a hydrocoral polyp, one of the cnidarian polyps, and they will suck out the contents. And you notice that there's essentially no body to this thing. All the legs seem to connect up with this one thin strut at the top. And so you could logically ask the question, well, where are all the guts in this thing? It's got to have a stomach. It's got to have gonads. It's got to be able to process all that food. So they actually extend down into the legs, which is kind of interesting. And there's an interesting series of studies on, how they move blood and oxygen and so on. You can see a couple of yellow dots in the front there. I think those might be, I'm not sure if they're eye spots or if we're seeing some of the ganglia in the brain. And notice that little nubbin at the back. That's basically all that's left of the abdomen, highly reduced. And then the two kind of leg-like structures that are coming off from underneath all of the other legs that are curled at the end, those are ovigerous legs, and so they would carry the eggs. In fact, it's the males that carry the eggs after the females have laid them. So really nice view. And I can't resist saying that I think the white stick on the right is our first carnivorous sponge. And maybe there's our second carnivorous. No, that's not a carnivorous. Never mind that one. And the sea spider is walking by that feather-like hydroid and is taking us right into the sponge that I wanted to see in the first place. So thank you, sea spider. Yeah, I don't recognize this sponge at all. Another one of these bryozoans underneath this sponge. This is kind of interesting. I sure wish I knew what I was looking at. Is it a sponge? You can see that it's got this branching structure and it's almost like a parasol above it. Now I'm thinking it's a tunicate. Yeah, if we can get a look on those holes, this must be a tunicate. Go to the left. I really want to see the structure above this branching. There we go. Huh. I don't know. This is a tunicate. Is someone on shore going to help me? To me, it looks like each of these units has some organs in it, those little sort of greenish brown structures that you see. And there are some very large openings that look like openings into an atrium. Yeah, Rian Waller's agreeing that it looks like a tunic. It's very floppy. I have never seen anything like this. So to really help, we need to go over to the left. It's not the branching I want to see. It's the openings on the top of this like parasol that I want to see. Thanks. So that's a good view, yeah. So one thing, Shora, to keep in mind, the camera does not pan left and right. It goes up and down. If we want to look left and right, the whole ROV has to move left and right. So the pilot sort of, we zoom out so you get a better context of where we are. Then we'll move the ROV, which is happening right now, and then we'll settle down again and get a good look. Huh. Yeah, Mary Bylan is saying it's like a parachute. Very interesting. Asako agrees, Tina agrees that this is a tunicate. Wow. I don't know that we have any tunicate biologists with us, so I don't know how unusual a thing this is. But as I've said, I've seen an awful lot of deep sea video, and I've never seen this. So tunicates are in the phylum Chordata, so the same phylum that the vertebrates are in, and this is colonial. So each of these sort of bubble-like structures you're seeing on this parachute-like or umbrella-like morphology is a separate zooid. Each of them would be creating a current with cells on the inside of those structures that have cilia associated with them that beat and pull a current in, and they each have a series of filters that they filter food out of the water column, and then they transfer it into a gut. So pilot, I'm sure you can anticipate the next comment I'm going to make. And not just because of the tunicate, but the white branching thing at the end is that bryozoan again. So if we could take a chunk of that hard coral, it's going to be, I just don't know, because that hard coral is so brittle. What I'm afraid of is we'd reach out to grab it, and the whole thing shatters and falls, and we don't get it. So much as I'm interested, I just don't know how easy this is to do. And it would be really nice if some of our European colleagues could tell us, is this a sort of thing that you would see on some of the, the Lophelia reefs and so on in the Northeast? I'm not as familiar with the Eastern Atlantic species, so this could be something that's seen fairly frequently in the East Atlantic, but certainly not in the West Atlantic. Yeah, so pilot, so here's the thing. What I'd love to be able to do is sort of grab the more towards the substrate behind where that parachute thing is, but the colony that is on is really brittle. It'll shatter. So I think you'd have to, I'm not sure if that means a fast grab, insofar as we can grab anything fast, or a very slow closing of the jaws. And I think it's okay if some of that stuff tumbles down, just not the part that has the parachute and the white fin at the end. So there's your challenge. The gauntlet's been thrown. Correct. Well, the parachute isn't super fragile. That's actually very flexible, but what it's growing on is very fragile, and so I expect you're going to see some degree of that collapse. It's brittle. And what I said to base, if you look just under the parachute, you see sticking straight down to 6 o'clock, there's that white fan, and that white fan is also part of the critical reason I'm asking for this. So you would need to get the manipulator behind that umbrella and fan. It's okay. I've seen enough from over here to know what it is. Thank you. Mm-hmm. So pilot, my apologies, just a request on shore. Can we put the lasers on it just to get a sense of how big it is, unless you already have a sense? Thank you. Okay, so it's not all that large, I don't think. I think I heard you say coming from underneath. I think this is growing from the wall behind us out towards us. And I'm wondering if coming from the top, but, you know your business. I'm not going to guide you anymore. Going dark. Yeah, I don't think that's going to go anywhere if you're able to get the hard coral behind the parachute. So away from us, closer to the wall. Stand by. No worries. That's exactly what I thought would happen. So, yep. Yep. You absolutely have the critical bits. So far, so good. You still have the important part. All right, terrific. So that's excellent. So we got the primary target there, which was the tunicate, and all the other corals basically have been returned to the sea floor. So good deal. Well done, pilot. That was stressful. Understood. All right, so for some context, we're just making a pilot change here. We are at 1,723 meters. You're hearing us if you're tuning into this live stream. The voyage to the Ridge exploration team on board the NOAA ship Okeanos Explorer. We've been out here for a week now. This is our sixth dive on the Mid-Atlantic Ridge. We are currently about 180 kilometers north of the Azores, and we're diving on a feature that's in the axial valley of the Mid-Atlantic Ridge, and it's itself a smaller ridge feature, and we're diving up the east-facing slope, diving towards the west. And we started at about 1,750 meters depth, so we've only gone up the slope about 30 meters. But we're finding just tremendous amount of material, really interesting biology to look at. On shore, your microphone is open. I think that might be the ECC. Yeah, 1,759 meters was the depth, and that was, let's see, 12, just over 2 hours and 15 minutes ago that we landed on the bottom. So our diversity and abundance continues. I see an Iridogorgia colony, Leopathes, Acanella bamboo coral, several different kinds of sponges, vase-type sponges, tube-type sponges, encrusting sponges. Most of them, I believe, are glass sponges. You can see how steep the slope is here that we're moving up. Some plexaurid sea fans, Paramaricia, Bathypathes black corals. And then, you know, a lot of the structure that you're seeing is this stony corals, scleractinian corals. The identification we got was Selenosmilia, was the genus. And the darker stuff that it's all growing on is dead Selenosmilia. So there's all kinds of coral rubble here that's on top of the basaltic feature, this ridge that's down in the mid-axial valley of the Mid-Atlantic Ridge that we're climbing here. Also seeing some of these sort of bottle brushes are Chrysogorgia, and Trisopathes is that sort of brown branching one that's off on the left. It's a thin, wiry Leopathes in the lower left. It's kind of interesting that the central part of the ridge that we're just climbing here looks really dense, but as we look off there, we're looking towards the southwest is the view that the ROV has now. Now it's turning, so you're looking almost due west along this slope. And so most of what we're seeing is on the sort of the crest of this ridge that we're climbing. It's a Parantipathes black coral that we're just climbing over. I think this is another Iridogorgia on the foreground. Even for me, who's not a trained geologist, I can see some of these pillow-like features that have cracked open and we're seeing on end. It's really easy to see that in this pile that Ashton was describing earlier as, you know, sort of pillow lava flows on top of pillow lava flows on top of pillow lava flows. So that's what most of this feature that we should be climbing today is constructed of. We're basically at the home of where the Earth's crust is born and new seafloor is formed all along the seafloor spreading ridges that encircle the globe. And so this would be the one that's in the middle of the Atlantic Ocean. And basically the plates are pushing apart from this point to the west and to the east, and so pushing North America further apart from Europe and South America further apart from Africa. And as they move apart, new crust is formed in between, and we are basically right in that zone, that rift zone. And great, Ashton is back now to tell you about it. We just had a chance to look at all the rocks, and now we're going back to the biology, so perfect for you. More or less.
Well, as soon as you guys leave for lunch, I'm probably going to get another rock. So.
Well, there was a lot of opportunity right there.
Yeah.
So I will get my lunch. Enjoy, Ashton.
All right. Thank you. Thank you. And can you put up the sample? Yep. I'll log the C-tube portion. Yep. Yep. Thank you, thank you. And just for my own awareness, are we finishing up a ship move or? Okay. And if you are just joining us, we are on the Mid-Atlantic Ridge, approximately 180 kilometers north of the Azores, north of the island of Corvo, the northwestern island in the Azores. We started our dive at approximately 1,800 meters depth, and we've been moving up this upper ridge of a normal fault that's west of the main spreading center of the Mid-Atlantic Ridge and just north of a fracture zone known as the Kurchatov Fracture Zone. We're at a depth right now of about 713 meters below sea level, and we are at 40.662 north, 29.385 west. I'd love to grab one of these fragmented pillows, but I realize it's down below us, so it's probably difficult. What's the plan? Is it to transfer, or is it to transit through water column to the next slope, or? Are we looking horizontal, or are we looking kind of down? Okay. Yeah, just trying to figure out. Okay. Okay. Yeah, I mean, it looked like there was outcrops beforehand. There'll be outcrops again, so it's not worth futzing around if you're going to jump over to the next slope. No, I think that's fine with me. Yep, no worries. Sounds good. Thank you. The tops are usually better just because if we do have to pick up a piece of rubble, then, if we're picking it from the top, we can kind of infer that it came from there, whereas at the bottom, it could have come from anywhere on that slope. But then, this is a good-looking outcrop just from the standpoint of you can still see some of those pillow structures, so those kind of lobate cross-sections like the one on that bottom left. And so, we're kind of looking for pieces that might have broken off something like that. But, we saw something like this earlier, so, yeah. Yep. If there's rocks ahead. Yeah, it's funny, when we did that dive a few dives ago on that axial, volcanic ridge, which should have been one of the youngest features in the axial valley of the Mid-Atlantic Ridge, I expected it to look more like this, these more fresh-looking pieces of basalt and kind of clearly identifiable pillow lavas. And that volcano I expected was probably only 5,000 to 25,000 years old at the oldest, but it was just so sedimented in biogenic crust and sediment. Here we're at a place that's 500,000 years old, and there's a lot of exposed basalt. And I'm looking at the shore-based scientists in the chat, and I've got the word rat tail, asteroid, and starfish, and I don't know. I know that's a starfish.
Hey, this is Chris Ma.
Hey, Chris, how's it going?
Hey, so this looks like a neat gony asteroid. I know they look fairly generalized, but I'll tell you, the last few times that we collected sort of these generic-looking gony asteroids during the last two Okeanos expeditions, they each turned out to be new species.
Oh, wow.
And so if this one can be collected without any imposition on other priorities of this dive, I can't guarantee that it's a new species, but by looking at it, it doesn't conveniently fit into any of the species that I'm thinking of. I mean, especially at this depth.
Yeah, let me chat to the ROV team. We actually just put in a ship move about 2 minutes ago. So we'd have to move the ship back if we wanted to collect this.
Okay.
Yeah, so we're no matter what, we're about to start moving away from this. This is what happens when Scott goes to get lunch. It puts me on the hook.
No worries. All I have to, every time this comes up, I have to ask.
No.
But if we can, then we can. If we can't, we can't.
No. I appreciate you chiming in, Chris. That's great. And I think once we complete this move, we've got a location here in the log, and then we can reassess.
My thanks.
And so if you're watching from shore, one of the live feeds we have on camera 3 is our quad cam. And in the upper right of the quad cam, you'll see a map of the bathymetry, including depth contours, that the ROV team is using to navigate. And you'll see that we're coming up on probably the first of 3 little topographic or bathymetric highs. So these first 2 are kind of just the local highs, and the last one is pretty much the highest point on this ridge. You know, if we can head to, or slowly make our way towards maybe that transition between where you have all those corals and then kind of those big blocky pillows, that might give us a little bit of both worlds. And it's hard to tell whether this is just much more heavily sedimented, or, yeah, these look much more like a talus slope or a rubble pile of broken pillow basalts that have come down this little chute. You can see they're much more angular. They look like broken fragments. And one of the things that's interesting to me about this transition is a lot of these corals and sponges we see, we tend to find them anchored on these hard substrate that we've been seeing previously. But there's not much on this slope, although there is hard substrate available, which means that this might actually be kind of an actively failing area. So rubble and talus keeps coming down this slope and maybe wiping out any chance for organisms to get well established. And you can see just on the other side of this talus slope field, you see the increase in those communities. And so this is one of these little shoots that the biologists usually tend to avoid, given the lack of organisms that colonize it. And so I imagine as we start coming up this slope, we'll start to see more of those in-place fragmented pillar basalts and less of this talus pile, and then maybe a blanketing again of these biological communities.
Okay, well, I get the impression the moment is lost. So thanks very much. If we see it again, I'll be watching. Hopefully we can reacquire another one.
Thanks, Chris. Ken Sulak was chiming in and saying that one of those fishes we just saw is a rare species, Atratodenteles frix, not ever imaged in the subsea previously. Yeah, so RV, is it easiest for you guys just to kind of follow that ridge ahead of us? Okay. Can you look to your bottom left, that little outcrop of rock? And you see that rock kind of in the middle with a shadow under it? Yeah, the lasers are just to the right of it. Yeah, could you see if that's snaggable? Yeah, so as you can see, we're kind of on this localized high. We're not at the base of a debris pile, so we know that the rocks here are kind of in situ. Now, this whole feature that we're on a broader scale has been uplifted as part of a normal fault, or actually the stuff more downward of us has been down dropped. But what we're looking for are these nice pillow basalts that have been broken up through that faulting process, but not part of a larger debris field. And so one of the things we'll try to do is see if we can grab a sample of that. Yeah, let's give it a little nudge whenever you're ready. And now, one of the things I found really interesting on this expedition so far is most of the cruises I've been on have been focused on the geology. And so when we're looking for rock samples, we're looking for rocks, and we don't necessarily have biologists on board to study things that might be on the rock. But, if we take this rock with us, there might be 5 or 6 different species that are living on it. And so that'll give our opportunity to our biologists to study other species of opportunity. Yeah, let's grab it. We put one rock in the port box, but if we grab this and take a look at it, it looks like it's going to be different enough for me where we can put it in the same box for my purposes. Yeah. Yep. Yep, and just as long as we get a nice glamour shot of it. Okay.
Did you get a rock earlier?
No, not yet.
This is the first one.
Yeah, that's great for me.
Wait. Oh, yeah.
Yep, this is great. Yeah, you know, it's a little different from the biological sampling from the standpoint of, you know, on a geological dive that's maximizing geological samples, you might get 30 to 40 rock samples. And so we really try and cram them all in the same space. We don't have to necessarily separate them. And it's all about finding, you know, a good photo of it where we can tell the different part of it. So one of the things I'll do is I'll take a picture of one of these sides, which I already did, and that'll allow me to compare it perfect to the other ones. And that way we can fit, you know, we can fit 10 rocks in the same box. We just got to make sure that Scott takes longer lunch breaks so I can get those 10 rocks.
It's a pretty good lunch today, so
Yeah.
I gave you some time.
Well, it was my favorite game, lunch game, which is the game of pizza. And you play by eating as much pizza as you can, and the winner is the person who eats the most pizza. And the prize is great because the prize is more pizza. Did you not have the pizza?
I did not.
No.
Sorry to disappoint.
Yeah.
I went with the vegetarian sausage.
Yeah.
Is that a snail? A large snail right there?
Yeah, someone, let's see, Tina called it out in the chat. Thank you, ROV.
So port forward, yes.
Yeah, and so if you're looking at this, this looks like a large portion of what was once a nice conglomeration of pillows, nice pile of pillows, not conglomeration, excuse me. And you can actually see that the fractures almost look like they're radiating out from maybe a central point of it, just to the bottom right of the lasers. And so what I'm hoping is that a portion that we got is from along those fracture planes, and so it came from this outcrop.
The snail is up and to, it's above the left laser, just in the crook of the rock. How come it looks like it's, I guess it's still there.
Yeah, it's still there.
There's a little U in the rock and there's something white at the base. I think that's the snail. Yep. It must actually be mostly on the other side of the rock. I am wearing the wrong glove. Looks like a bucynid type, whelk type snail. That's the one. You can see those ribs in each of the whorls of the snail. And we're not going to see its head. It's tucked down there, but this is good. That's a good view. I don't think we'll get anything more from lingering on it, so I'm good when you're ready. See another one of these stalk crinoids here with the stalk just covered in these hydroids. It's really interesting. Yeah, so this is what Ashton was just telling me off mic, that there's these local highs, these ridges where you get all the suspension feeders, and then next to them, I wouldn't call it barren. It's not barren, but obviously there's a lot lower density of these suspension feeders. So I guess it's something about the dominant current and the way some of these, I mean, the marine snow is moving from right to left, but the way some of these whips are oriented and the fans, I would have thought the current was going the other way, but maybe
yeah,
not.
To me, there's actually looks like maybe 3 different environments. There's the one kind of on the central right of this ridge that looks really rich with sponges and corals. There's one on the central left, which looks somewhat less populated with macrofauna. But then there's also the kind of far right, which looks much more angular talus.
Yeah, and that's the chute for sure.
Yeah, the debris field,
Yeah. It's lower. Looks like there's more sediment accumulating in there.
And so it's amazing. It's a small area, but with at least 3 very different types of local environments.
I can already see the slope above us that's moving up. The contours is chock block full of these suspension feeders. Let's hope we see the Chimera again, because Ken Sulak has provided some additional information on the lateral line system. It'll be interesting to tell you about in context of actually seeing the fish, which he identified as the genus Hydrolagus. I think that might translate to water rabbit. Lagomorph? I think lagomorphs are rabbits.
The only thing I think about when I think about watership, or when I think of water rabbits, is the book Watership Down.
Yep.
And then I start getting a little sad.
Yeah. Let's not go there.
Yeah.
I think I see an Enelopsamia, that sort of radar dish-like yellow fan that was on the far left. Let's see, what's our, so we were kind of looking southeast there, and now, northwest. I was looking at the backside, and now we're pointed almost due west, upslope. One of these large platy sponges. Yeah, there's that very bright yellow. If we can get a snap zoom on it, I think I know what it is. I think it's Enelopsamia rostrata, but let's just verify that. Yep, sure is. So that's another scleractinian. So all this rubble that you're seeing and some of these live colonies we've been identifying as Selenosmilia, those are stony corals, belong to the order Scleractinia. They're the ones that in shallow water build the massive reefs. It's a different group from all of these other octocorals, the sea fans that we're talking about. So this is a different genus and species from that one that's right behind it that's more beige in appearance. And you can see that it grows kind of like a sea fan, except it's extremely rigid, very brittle. And that's a nice healthy one. It's alive. All the polyps are facing, I guess they're facing into the current.
And is that fan shape pretty standard, or do they tend to be also three-dimensional?
It is for that species.
Yeah, for that species.
Yep, absolutely. Yeah. And not only that, they always have the polyps just on one face. Very characteristic. And it's one of the reasons why, for a change, there's an easy one to identify. The shape, the position of the polyps, and the color. Thank you very much.
You're welcome.
I meant the coral. Thank you for making my life easy. Beautiful view of a dead bamboo coral there behind. I don't usually say that about bamboo corals, but we've talked a lot about nodes and internodes, and you can see that very clearly. That is an internodal brancher back there. So the white is the calcium carbonate, and there the lower part of the colony still has its tissue on it, and the black is the protein-based part of the colony.
And so when that continues to weather, will that break up along those protein boundaries? I mean, what does that look like in terms of detritus?
Yeah, that's a good question. That's entirely what I would anticipate.
Yeah.
Most of the ones that I've looked at closely or I've seen in papers, there's a thin rod of calcium carbonate underneath that ring of protein.
Okay.
And the interesting thing about that is one hypothesis was, well, if you have all those protein nodes, they could flex at that nodes. Well, they can't really.
Yeah.
Because
It's got a rigid infrastructure.
Exactly. So I don't know what it's all about. One of the dangers of trying to attribute to every single characteristic you see in an organism is saying that it is an adaptation to X. And, well, we don't know what was happening at the time this evolved millions of years ago. We're interpreting what we see now. So you have to be careful to keep those two separate. Oh, Mike Vecchione is reminding us that there's a tidal component to the near bottom currents out here. So the moon does influence the movement of water down here in the deep sea. And so when we talk about currents, you know, it's what's happening at the instant that we're looking at it, what's happening on this daily and lunar cycle, and what's happening over the much longer cycles of the period of time that these corals have grown. And so you may get, you know, a whole lot of different motions and rates of motion in that time. And so I try to usually look at the corals as kind of averaging out for me what's the dominant current. And you can tell that in some of the fan-shaped ones and some of the others, the way they've sort of fixed into place. They should know what they're doing, even if I don't.
Well, you talked about the moon, so I'll bring it up. Part of my day job is monitoring the variation in the gravitational field over active volcanoes. And we get changes in the gravitational field depending on whether magma is intruding beneath the volcano. If there's more magma beneath the volcano, that usually contributes to more mass beneath that volcano and a larger gravitational field. And these instruments we use can actually measure the variation in gravity on the order of 1 to 1 billionth of what you feel under normal circumstances. And most of that motion or that contribution is from tidal effects, so it's from the moon. So if there's a full moon out, or if the moon's a little bit closer and you're feeling a little heavier, it's because you probably weigh about 1 billionth more than normal.
That's good. I shouldn't have blamed that last bowl of ice cream I had.
Or that third piece of pizza.
I didn't have the pizza.
I was talking about myself.
Okay. Ken Sulak has added some really interesting notes here, and there's one I want to share. Very early on in the dive, we saw an eel-like fish, and we got some really good video. We followed it around, and you may recall I said that it looked like it had been in some scraped up against maybe the coral and so on. And Ken says that that's a synaphobranchid eel, but it's a rare species. Let me see if I can pronounce this. Atractodendrocheles frix. And he says that eel has never been imaged subsea previously. So congratulations, ROV team. Another feather in your cap. That's very cool. This is one of the values of telepresence, is having these expert scientists from around the world who have made a lot of their own observations be able to share with us what we're seeing, what's unique, what's already known, what's worth collection, what's worth another close-up, and so on. Really valuable. Passing over several black corals here. Oh, so this yellow colony, this yellow fan, let's see, there's two of them. We've got a collection request for one of these yellow sea fans, and there are two different morphs, but I think I'm seeing two of the one they want to collect. One is just off the screen lower right. The other is right of center at about 2 o'clock, hanging over a bit of the rock. So if one of those is in a good position, we can collect, but I'm seeing so many of them that it doesn't have to be now. You let me know. All right. Yeah, let's go for it. Yeah, so I'm seeing there's all kinds of them. Yep. Yep, right under the lasers. Yeah, and the key pilot that I want to look for is when we get a close-up is to see, is there a brittle star on it? I'm not expecting there to be a brittle star on the version that we're looking for. Wow, look at them all over here. And, yeah, Manuela, I think if you're still in the, you are still there because I can see your name, so please let me know if this is the one that you're targeting. It's really hard with these plexaurids. They're pretty diverse, and some of them are hard to tell apart. Yeah, she says it is. Great. So we've got that confirmed. And so one of the reasons we're targeting this here right now is that, well, actually maybe you guys down in Horta, if you would like to tell us a little bit about the work that you've been doing in this area with the drop cams and your previous dives and, you know, your knowledge of the local community and why it is that, you know, this would be a valuable collection. I'm sure they'd rather hear from you than hear from me again.
Hi, Scott. Thank you for calling us. So over the last, let's say, 5 years, we were trying to have an idea of what are the benthic communities that live inside the exclusive economic zone around the Azores. And because the Azores and Portugal, we don't have a really large research vessel. We do have an ROV named Luso that goes down to 6,000 meters. But it's hard to bring the Portuguese ROV Luso to the Azores. So we were really dependent on cruises like this one to have some images and some samples of the benthic community living in the island. And the research questions that we are working with are different folds. It goes from the individual species ecology all the way to what drives the distribution that we see, why some species live in one place and why that specific species does not appear in another place with very similar environmental conditions. At the end of the day, all this information is being used to inform the regional government of the Azores in their maritime spatial planning and in the designation of new marine protected areas. As we speak, we are working with the government in order to identify the best areas that could be used to achieve certain conservation management goals. And the aim of the regional government is to protect 30% of the EZ by 2023. With 10 or 15 strictly protection. So all this information is being extremely valuable to inform those decisions. But going back to my story, so we were dependent on cruises like this one to do the explorations. And we have very good collaborations with our colleagues from the Netherlands in the NEOS Research Institute, with our French colleagues from Ifremer and Sorbonne University, and also with our Spanish colleagues from EAO. And when their vessels come to the Azores, we try to hitchhike and take the opportunity that they are around to do a couple of days of work that would fit our research agenda. But these developments are being really, really slow. And instead of kind of waiting for the new vessel to come, we decided to think out of the box and try to develop some imaging tools that could be fitted to our reality. And our reality is deep seafloor close to the island shore, small vessels, and also some vertical walls and also some lost fishing lines on the way. So after a few trials, we developed a system that has been extremely effective on taking footages of the deep sea. We called it the Azure Drift Cam. And because we think that this would be useful for other scientists around the world, we made that prototype available as an open access scientific publication where anyone can download. And if you go to the supplementary material, there's kind of a recipe book on how to build and operate this tool. So over the last 3 years, well, really 4 years, we were amazed by the amount of kilometers of seafloor that we could cover. We, as we speak, we are probably at around 600, 660 dives with our system. We covered, I can't remember on top of my head, but tens or hundreds of kilometers of seafloor. And now we have a much better idea of what lives where. But, of course, that this low-cost system came with lots of caveats and lots of problems. One is, for example, because we drift, we can't tell or we can't select where our camera system is going. Like your pilots, they do it magnificently. You select where you want to go, what species you want to see, and they magnificently put their ROV in the place. So with a drifting system, we can't do that. So sometimes we see things on our side and we can't really go back and have a look at what it is. So the other problem is that we can't take samples. And because we are doing a lot of explorations in places that haven't been visited before, it means that we can't put names on a lot of things that we see. And that's why we, Manuela and Carlos, are asking for samples of a lot of things that we have on our image catalogs, but it's like identified as morphotype number 25 or so. And it's really important for us, this effort of collecting samples, so we can go back to our catalog and change unidentified species number 125 by a proper name. And then all our databases are automatically updated. So this is really, really important work that what you guys are doing. So we are available here for questions via the chat or via email. But in brief, this is what we have been doing over the past years. Back to you, Scott.
Sorry. All right, sorry folks, we're doing weekly drills here on ship, so don't worry about the bell in the background. Copy that. Sorry, pilot, I was just trying to stay off comms while the announcement was going on. Yeah. So, yeah, beautiful cut, by the way. That was really nice. Suction is great, and we're all happy, and you do what you need to do. So, yeah, I think that was Telmo Morado giving that, and I apologize, Carlos, if that was you. Please correct me. That was a wonderful explanation. Thank you. Really appreciate it. And so, yes, hopefully now with this collection, we have one number that will be changed into an actual species name. So the pilots are going to slurp this sample so that we can maintain the remaining 2 bio boxes we have for larger samples in the event that that's necessary.
Now, when I was talking about rock sampling earlier, I was saying, sometimes it's okay to put the same rocks in the same bin because we can tell the difference. Do you ever do that with the biological samples, or are you worried about associates transferring from one to the other? Or?
Yeah, so we will occasionally do that with the biosamples, but you've just identified it. If we have reason to believe that this is probably a sample that has a lot of associates, we'd like to keep them as separate as much as possible. If it's something either we don't anticipate there's associates or we believe that the associates are on there are likely not to let go, and it's very easy to tell them apart, then we'll do what you did. Yeah, let's take advantage and use all that space. Part of it go in. Yep, and it's in a canister. Very nice.
Yeah.
It's a darn good simulation. Oh, man.
Yeah, so that was Telmo Murado, right, who was talking about the Azores drift cam, and I'm looking at the paper, and it's really beautiful imagery that they're able to produce from it. It's really a great system.
Yeah, no. What I was saying is it was your option to choose between the two fans, so we are good. We're happy with the collection. Thank you. Yep. Getting a lot of interest from shore and our sponge biologists in some of these sponges, so I'm getting some feedback to find out which of them would be particularly interesting for a collection. Probably the answer is all of them, and so we'll try to narrow that down. She notes that most of the descriptions of these species from this area are from the early 1900s, and this is a common problem for a lot of deep-sea biology. They're from the 1900s, and they're based on dredge material. That is, somebody was from a ship dredging a device that would scoop up rock, sometimes be for for fish and soft sediment organisms, but you wouldn't drag that kind of a trawl through an area like this. And so this would probably be a rock dredge that would get the sponge. So she says being able to do proper taxonomy and genetics to match them with the original description would really help the understanding in that field to match what did they describe in the 1900s compared to what does it actually look like when we're down here with an ROV. Sorry, just typing some notes in the chat room. Figure out our target for some of these sponges, and one that's of interest is the large white frilly one. All right, the gramophone or funnel one with the thin walls. Thank you. That's helpful. This is the bright purple Trachythela or Clavularia, the octocoral that overgrows various kinds of substrate rock, old coral skeletons. I tend to find it more often on skeletons of other organisms, either sponges or corals, than I do directly on rock. And there's a whole lot of coral rubble here, encrusted to some degree, turning it black. And you see it in the upper left, there's a couple of the orange coily colonies. Those are probably a Phanosticopathes. Actually, there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. There's at least 10 colonies of those black coral whips right in that little patch of exposed hard substrate. Isn't that interesting? Not right here. I think we've got a good handle on what we're looking at right in front of us. It's like a Chrysogorgia peeking out from the back of, I'm assuming, well, never assume. I don't know if that big black branching thing just going out on the right is an old dead sponge or an old dead coral, but it's got a variety of things growing on it, including a Chrysogorgia. That colony in the upper right is Trissopathes, black coral. Kind of fleshy polyps, lots of branching. Interesting color compared to a lot of the other black corals that we've been seeing. Pilot, I can tell you one of the things that we're looking for is a white sponge that grows kind of like a funnel. So I'm going to keep my eyes out on that, and if we do see one that's in an appropriately positioned...
Is that one at the top?
You know, that might be one here, but I'm not sure. I will point out as we're looking around, that one on the top, you know, you'd have to go in over those other corals. There's a sea star feeding on one of these whip corals in the center. Oh, we can actually keep going to the left. Don't worry about the sea star right now. That's a behavior we've seen many times, but this might be the sponge we're looking for. Let's see.
These are not the sponges you're looking for.
Yeah, well, that's exactly what I'm waiting for Joanna to tell me. Okay. All right. I do see off to the left the first, I believe, Metallogorgia. I don't think we've seen any Metallogorgia so far in this cruise. And pretty sure that's what's down the slope. That large one is the frilly one. Yeah, so I think it's right in front of you, Fernando. Yeah, no problem. The sponge that you have been stopping in front of here, that is the collection ticket. Right in the center bottom. So just again to clarify, Joanna, this is the sponge that you're referring to? Yes, okay, good. So yeah, we'll get some good video of this, and then we'll probably need to, you know, cut or peel a chunk out. There's an Okeanos bamboo coral behind the sponge. Just getting a good close-up view of this. No, I'm just relaying to shore basically what we're doing. And I'm trying to get some input from shore to get a sense of, is this going to be kind of crunchy or is this going to be kind of soft? Just so you are aware before you grab it. An interesting hydrozoan growing down below, I think, that sort of bluish colony, lower left. So this will be crunchy in that it is a glass sponge, but flexible enough that it'll be easy to move the manip through. Thank you. Yeah, so I would say a little bigger than the size of my hand at least. And I don't think it matters where you get that piece. No, I do not think so. I think you'll go right through it with the fingers. Yep. Just a little bit more than that. Thank you. Yep, that's good. Yep, that'd be very good. And you might not have to entirely close the jaw. That's what I'm being asked. So you don't crush it. Yeah, just see if it twists. Yeah, looks good. Beautiful. It looked pretty soft to me, so yeah, I think I'd tuck that hand down in there. Okay. Let's just hope there's no current. Oh, that looks good. Oh, you grabbed it. You wanted to give it one more little shake. All right, nice. Nice collection. That is in the starboard inner bio box. Paying a lot more attention to my starboard port today. Survey 1, we called that Hertwiggia. Upper left. All right. All right, good stuff. Nice collection. Can we just look towards the left? I want to verify that I did see just slightly downslope a Metallogorgia, and I'll try not to send you down there. Just we'll have a look over. And it might be too far downslope. I saw it before. Let's not worry about it. We'll see it. We'll see more later. I am seeing an Oreo out there. Also named the false boarfish. That's the fish off in the distance. Pilot, you don't need to worry about it. But the reason I'm bringing it up is that Ken Sulak provided some more information. And he says that the false boarfish, some species off Australia and South Africa, get large and are fished commercially. The mouth looks small, but it's highly protrusible, so it can stick it out. Oreos are associated with deep reefs and rocky drop-offs. Oh, I got an auto-refresh, so I lost all that. They feed on small invertebrates, and he suspects they also nip coral polyps, or they are biting at amphipods that are on the corals. And Chris had noted a white sea star, and I apologize, Chris. I was looking around for it, and I don't see it now. So we can continue our move up slope. Are you able to just zoom straight into the center? There's something, a brown circle, brownish red circle, just below the lasers. Yep. Oh, I see. Okay. It's an anemone that's somewhat contracted. That's fine. Thank you. So since the time that we arrived on bottom, that was, 4 hours ago, 4.5 hours ago, 11:44, almost 4 hours ago. We were at a depth of 1,759 meters. We are now at 1,684 meters, so we've come up about 100 meters on the slope. We've been seeing this kind of density on this coral rubble. It's really quite remarkable. High density, high diversity. Chris Ma noted as we went over that bubblegum coral that there was one of the large sea spiders on it, Coloscendus. We did get some really good video of a Coloscendus earlier today. So that would be the second one. It wouldn't have been upslope that fast, I don't think. Or maybe Ashton will tell me, no, you are going that slowly.
I would never say such things.
Thank you.
I just never thought geology would be the comparatively fast-paced action science that it is.
Yep. Can we, are you able to turn to the left, pilot? Do you have the tether? If you can continue to turn to the left, below the white sponge, further to the left, there's some fans, sea fans over there, and I see something tucked underneath them. It's kind of beige in color. I just want to make sure that's not an octopus tucked in there. Looking right there. It's probably more like the back of an anemone or something, but it's certainly an odd texture. It's yep. It's you're going towards it. It's if you I see the lasers. So it's almost above the lasers, but at the top of the screen.
It's the eyeball of the circle of yellow coral.
Exactly. Sauron or something. Is that what you're telling me?
There you go.
Yeah. I no longer think it's an octopus, but yep, that's what I'm looking at. Oh, another anemone fooling me. So again, this one's kind of tucked underneath there. We're seeing the side of it really bulged. We're looking down. I think that opening above it is the osculum of the sponge. All right, that's good. Thank you. Sorry to get Mike all excited there. I just saw something, kind of pink tucked under that coral. Another Iridogorgia in the top center. So several of these things, if you're just joining the internet live stream now, we've been climbing for about 4 hours now. We've seen many of these corals and sponges, and we've had some pretty good views. But as you see, just as we're turning here now, the sheer density of material is such that we wouldn't get everywhere if we tried to look at every one. So we're trying to get, some good looks so we have an idea of what these are, and then someone can count them later. I will say that perhaps the students in my lab are going a little cross-eyed today as they look. In theory, they are meant to be annotating all the organisms that we see on the bottom. And there's far too much to be able to annotate everything, but we'll be able to say, something like high-density coral field and try to pick out as many things as we can. Really large colony of a Leopathes black coral just passing off to the upper right. And so now we're back into one of these chutes that Ashton was referring to before. You see there's far fewer organisms. Partly that's because probably there's more tumbling of rocks in this chute, and so stuff wouldn't have a stable substrate for a long period of time, but also maybe because it's down below these two ridges on either side, and so it's not experiencing the same water flow.
Yeah, and I think it's interesting, at least on the other side of these two ridges, the rock that we're seeing, the kind of the exposed rock surface looks much more intact. It's highly fragmented and it's broken up, but it looks like the rock that was in place, and this clearly looks like a giant pile of rubble.
Yeah, stability is important for a sessile organism that's going to be growing in place for many decades or hundreds of years.
So what would be interesting to me from the geological side is getting to the top of this chute where things are actively falling off, because that's a really good place to stay off of rocks.
Yeah.
Probably not a great place to live, though, if you're a biological
perched
organism.
right on the edge.
Yeah.
Yeah, I too would like to see the top. If we can zoom in, it looks like right in the middle there's a white thing. That might be one of these white sea stars that Chris Ma was referring to before. Just to the right of, yep, that's the one. There we go. See if Chris is still, looks like he's still in the chat room. It's a little bit far from us, Chris, so it's going to be hard to get a really tight zoom on. Not 100% certain, but maybe something like a Terranaster or something. I feel like I'm seeing a vent in the center of it. Maybe not. And is that our maximum zoom? Okay. Oh, good. Chris does say Terranaster. So I think, Chris, is that the quote-unquote slime stars? Yeah, you can see that little opening at the top, a little sort of window or funnel. As I've heard Chris describe it, it's kind of like there's a false skin on the upper side of this sponge. And I think you can, I'm guessing it's the same thing that you see around the legs, where you see some of the spines go into this thin membrane. So it's almost like it, to me, if I understood Chris correctly, it can inflate this upper surface above the actual dorsal side. I guess it's not truly dorsal since they don't have a dorsal and ventral, but functionally dorsal. You can also see all the underlying spines inside that membrane. There's a lot of ossicles, calcareous ossicles, and spines that make up echinoderms, and really easy to see there in that sponge. Thank you. Yep. Chris is no doubt cringing over all the things that I'm saying, pounding his desk. And there he is.
Hello.
I knew I could get you to get in. Hi, Chris. You're going to have to do that from memory.
I need to. The phone has an auto dial. This is kind of cool because I've been studying a lot of the papers that the Russians have done in this area. And I didn't realize this earlier because I hadn't seen one that was in good shape. But this looks like one of the new genera that was described by Anna Dillman and her colleagues. Or rather, it was described by the Russians earlier, but then later figured in detail by Anna. I think this is something called a membranaster. And I think this might be one of the first times we've seen it alive. I'd have to go back and see how much of the other abyssal video had something similar to this, and how widespread it is, because a lot of these deep sea animals, as you know, they don't appear to be limited to one ocean basin, especially at these depths. So, it belongs to the same family as the slime stars, the Terrasteridae. Part of me would love to find out if all of the representatives of this family produced mucus, and, I'm sure firsthand experience by anyone unloading it would be a fun video experience, but I'm not sure I would put that through anyone. Anyway, but it was just very exciting because, like I said, I think that's like the third genus of Terrasterid that occurs down here, and I don't think I'd ever seen it before. So, we have yet to see Hymenaster, which is the other the other one with the gelatinous surface.
Hymenaster, is that what you said, Chris?
But yeah, there's definitely. I'm sorry?
Did you say Hymenaster? That's the third one?
This one, the one we just saw, that's the third. That's the big gelatinous one.
Okay.
The one with the big kind of pinkish, purplish, fleshy cover.
Yep.
Yeah,
okay.
that one will be called Hymenaster.
Okay.
Yeah, but this one I think was called Amembranaster. Again, these things are complicated animals, and looking at them would always be the best way to tell for sure. But
yep.
Perhaps Anna is listening or watching, and she can take a look. But
Chris,
there's definitely.
That's the first time I've heard that term, Amembranaster. That's the first time I've heard that genus name. So thank you for that. And I'm just wondering, did you say I was typing something in the chat, so maybe I missed it. But so, what is the function of the mucus that it's producing? Does anybody know?
No, I mean, at this point, it remains kind of an anecdotal experience.
Yeah.
I mean, so here's the thing, I mean, other than that first account of Pteraster tessellatus, the shallow water species, back in I think there was one paper initially, and then there were several papers afterwards by what's his name? But in the 70s, when marine ecology was a big experimental paper thing. And then it just kind of stopped, and no one knows what they do. I know from personal experience that Hymenaster produces a copious amount of mucus when it's collected, but that remains unpublished other than on the pictures that I've shared on Twitter. So in terms of, everything from the chemical composition to whatever, function it might have, I mean, it's presumably defensive. But I've talked to people who tell me that not every pterasterid that they've collected has produced mucus. So it's not clear to me how many of them even have mucus production, or if it needs to be stimulated in a particular way, like if they were poked or provoked in some way. So I mean, it could have it could be that some taxa are just better equipped with mucus cells than others. But I mean, it's one of those things that's fairly difficult to test, as you might imagine, because I think the original experiments literally just provoked it and then they threw a bunch of animals in with it and saw how many of them died from the toxicity. So, as far as, experimental data on the function of the mucus, and most mucus has a biological basis in that it's some kind of glucopolysaccharide. It's very difficult to say what it does. So, I mean, because, that's a very versatile substance, but the original papers suggested that it was defensive because it does tend to kill everything that's around it, or at least not kill, but it clogs up a lot of the gills and such. So, but the deep water ones don't seem to. I mean, I don't know how often they're provoked or how often they would have a cause to use it. So I would imagine that has a role. But Hymenaster and all of the pterasterids at this depth do some bizarre. There's some bizarre architecture. It's not clear what they do. So, that's why I've always made this point when we see them. Observation and ecology and their interactions with other animals is probably the most valuable thing we get out of Okeanos Explorer, other than the actual specimen of describing a new species and such. So, anyway, thanks very much. I see you're homing on a coral.
Thank you, Chris.
I'll catch up with you guys later.
Yep,
Bye-bye.
appreciate the explanation. So I'm pretty sure that was Jason Isis type bamboo coral J-clade. So thanks for that, Chris Ma from the Smithsonian Institution, sea star expert and knowledgeable about many echinoderms and many deep sea organisms, and always has some fascinating stories to tell us about them.
Have you seen those dark whips to the right?
Dark whips to the right. Yes. In fact, we collected some earlier today. That's the interesting plexora that has the, yellowish skeleton with the purple polyps. One of the things I am looking for, we passed over, and I wish I had said something at the time. No, we can keep going. I'll wait for the pilots to talk to Navigator here. Pilot, are you able to go to the left, or are you restricted to going to the right? I'm looking over in the distance. I see a colony that is a long stalk, and then there's a poof of branches at the top. That's what I'm looking at. Thank you. Yeah, so everybody, if you can keep your eye out, we passed over a sponge that I thought was quite purple in color. And it's... Yes, if you could turn the lasers on, it's, yep, the one that's coming into center. Right, so everyone also be on the lookout for bulbous purple or deep purple or purple red sponge. This one, yep. So I already know what it is. It's just, I think it's the first one we see in the expedition, so it's worth just pointing out. So this is another member of the octocoral family Chrysogorgiidae. It is the genus Metallogorgia. Almost certainly it is the species Metallogorgia melanotrichos. And 100% it's going to have one brittle star in the middle of those branches. You know what happens when you say 100%. If it doesn't, it'll be the first one I've ever seen of the hundreds and hundreds that I've seen that doesn't have one. There's a very interesting host-symbiont relationship between this brittle star and this Metallogorgia, where all throughout the Pacific and the Atlantic, we have never seen a living Metallogorgia that doesn't have that brittle star. As the pilot's getting in a position for a close-up here, I think I'm also seeing in the upper left corner one of the pom-pom anemones. Anemones that have deciduous tentacles, meaning they can drop their tentacles when they're disturbed. So the interesting thing about Metallogorgia is you see that there's no branches on that stalk. All the branches are at the top. However, what we have learned through past observations, collections, and genetics is that when they're younger, they do have branches along the stalk. It's just that they drop them as they get older. So I'm seeing two things here. The brittle star is definitely in the center there. You can see its arms all tightly wound. But I'm also seeing there's a cluster of eggs that's been laid, in this colony. I suspect that's a fish that has laid those eggs based on past experience. So one brittle star, you see its arms are all wrapped around, but those arms are really long. We spent a lot of time unraveling those arms on other colonies. And as near as I can tell, they extend to all parts of the colony. And you see there's no other organisms living on this thing. And so there's a combination, we believe, of chemical compounds in the coral tissue and this brittle star keeping this colony free of other organisms. But apparently it could not keep it free of a fish laying some eggs. That's my guess. That's good. We got what we need there. Thank you. And Joanna Xavier is asking if this brittle star only occurs on this species substrate. And yes, that is the case. This brittle star has never been seen on another coral or on the seafloor itself. It only associates with that one coral species, which opens up a whole interesting suite of questions on how the brittle star as a larva is able to find the coral, because we find it on the smallest of corals, so apparently it's found pretty quickly. Surely there's some kind of chemical signaling going on, but interesting question. Deep sea is a big place, and so to be able to find that coral must be challenging. Unless the larvae travel together, which seems absurd to say, but stranger things.
Why does that seem absurd to say? I don't know enough about larvae.
Yeah, because so what's happening, to produce a larva, you have to have the combination of sperm and egg. So it means first you have to have an egg fertilized by the coral. And, you know, I don't know. My impression is that usually takes place in the water column, but maybe not. It's possible that I believe those Metallogorgia colonies, they're separate males and females, so it's possible that the females hold on to the egg. Egg gets fertilized, cells begin to divide, and you start to get an embryo, and eventually you get a larva. Well, the sea star or the brittle star has to do the same thing.
Mm-hmm.
And so how do they time that, and then how do the two larvae travel together?
Oh, you're talking about the two larvae of the two different species, not necessarily like a clump of brittle spar.
Correct.
Okay.
I meant two different species, holding cilia.
Well, that
like
seems
holding
ridiculous.
hands. Thank you. I think I used the word absurd.
Yeah.
Pink sponge. Sorry, I missed the pink sponge. I'm seeing lots of pink octocorals. Ken says he's puzzled over what fish would deposit eggs on a soft coral, new to him. Ken, yeah, I'll check this out. I'm pretty sure, I seem to recall a paper about fish eggs on some sea pens. I was done up near Newfoundland. But we did collect a Metallogorgia back on the New England seamounts years ago. This must have been 2005, I guess. That also had an egg clump very similar to that. And I thought we had identified them as fish. But we'll check that out. Ken, just recently in my lab, we sequenced some fish eggs we pulled out of a sponge in the Emperor seamounts. And I was able to get some DNA sequence from those eggs and found out that they were from a snailfish. So that was interesting. Another sea star feeding on the coral down there. Yeah, Mary Bilan is saying that there's no rubble on this hill, none of the coral rubble that we were seeing earlier. But we're seeing some healthy-looking corals themselves. Are we getting up near the top where the tumbling starts?
I think it looks like we're probably getting near the top of the second of these topographic highs before the summit.
Yep. Yeah, we're climbing like a series of steps up. It's kind of like a side ridge on the main ridge. So the side ridge is moving, essentially, we are moving east to west up this ridge, and then the main ridge moves north-south. We won't have enough time in the day to move north-south, but we'll hope to get to the top of this east-west trending ridge. At least in this little part of this area we're transiting over, it looks like the stony corals are the dominant right here. Pilot, if we're able to go to the top to that big white fan. Thank you. Copy, pilot. Pilot, shall we then just zoom on the sponge that's ahead of us without you moving? Yeah, the cone one that's a light brown straight ahead. Looks like a little vase or sponge. This is vase sponge. I think this is, well, this is quite different from the one that we collected earlier. See if Joanna's still around and can say anything about it. Otherwise, in front of it, the lower right, that's the plexaurid that we collected earlier today. Most of what is encircling the sponge is the stony coral that's been identified as Selenosmilla. There's a couple of Acanthogorgia colonies in here, Anthothela. Joanna says this is a Asconema. She says it'd be great to collect. Well, it's kind of tucked in there. Not sure how easy that would be, but as we zoom in on the sponge for Chris Ma, I noted just off to the right, an urchin also appeared to be feeding on one of these stony corals. Not 100% certain the sponge is still alive, but Joanna says it's either Asconema fristidae or Norda zoriense. Yeah, so from a distance, I wasn't sure if this was going to be a corallium or another bubblegum coral. I now believe that it is a bubblegum coral. We've been seeing the red morph this morning, and this looks to be the white or light pink morph. I'm not sure that it's the same species or that it's a different species. I do know that, for example, Paragorgia arborea has red, pink, and white color morphs. So you can't always use color to determine species. But this one also has a number of the Astroschema brittle stars on it. Look at that's a great view. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, maybe 14 or so of these brittle stars wrapped around. So it's definitely the family Paragorgiidae, probably the genus Paragorgia. In this one, you see the polyps are extended. The red ones that we were seeing earlier today, the polyps were all withdrawn in those sort of bubblegum-like wad masses on the branches, but here many of them are extended. There's also a series of barnacles growing on there and some hydroid polyps. Yeah, that's good for science when video is satisfied. And when you're finished with video, before you move, can you turn the laser 1 and 2, 10 centimeters, so 10, 20, 30. That's probably 50, 60. I mentioned an urchin. We may have moved over. It was an urchin feeding, but I think we may have moved a bit too far. Not sure where it is. Yeah, there's the urchin on the left side. Just about even with the lasers. Keep going left. It's the white ball. Right there. Go see a hermit crab up on the top. I think the mouth side is on the left, but that must mean there's a branch that I can't see that it's hanging on to. Interesting. Also, I'm just noting this for the people on shore, that purple one to its lower left is, I think, the Victor Gorgia. There's an amphipod that's just swum in there. Chris Ma thinks that is Okeanos. Yeah, and he agrees with me because it looks like the mouth is on the left side, and that doesn't make sense. The mouth should be oriented toward the substrate or whatever it's feeding on. So we don't understand why it's oriented this way. For science, we don't need this angle. If you can move to the left, we'll look at that again, but we don't need to do a big, huge move if that's not possible. We're just trying to get a quick look in there, wondering, is it hanging on to something? Huh. Well, that's just weird. If you have more zoom, just take it. Yeah, thank you. Well, there you go. You're looking at the mouth. You can actually see the teeth of the Aristotle's lantern, which is not something we usually get to see here because usually it's facing down towards the substrate. So I don't understand why it's there if it's sort of fallen off a rock and it's just sort of hanging above the precipice. Okay, thank you, pilot.
Yeah, could it just be moving from one location to another?
Well, it usually moves mouth down.
Yeah. Okay.
I mean, it may have tumbled and its spines have gotten caught on the
coral.
The coral, yeah.
And if you're just joining us, we are on a topographic high, a bathymetric high, on the Mid-Atlantic Ridge, just north of the Kurchatov Fracture Zone, at a depth of 1,661 meters below sea level. We started this dive about 5 hours ago at a depth of 1,800 meters below sea level. We've been transiting up this bathymetric high, this ridge that forms the upper portion of the footwall of a normal fault just west of the axial valley of the Mid-Atlantic Ridge before the entire Mid-Atlantic Ridge moves about 20 kilometers westward as it goes south. And that westward transition is a fracture zone known as the Kurchatov Fracture Zone. We are going up to about 1,630 meters below sea level, so maybe another 30 or 40 meters to ascend. You're looking right now, you're basically just looking at the blue water column of the ocean, and that's because we're moving from one of these highs to the next portion of the slope. And the ROV doesn't really operate very well going downhill, so whenever we get to one of these highs, we kind of fly through the water column. That's a safer operation. And we are at a latitude of 40.662 north and 29.385 west. Oh, and there's some really nice, beautiful possible sheet flows that you see in that outcrop right there. You can see those nice kind of layers, almost like pancake layers of individual flows. And then you see some pillows intermixed in them, the more low bait features. But that's what we think about when we think about kind of nice, solid, thick flows that are flowing as sheets, especially right now in the middle of the camera. And you can see again, as we saw before, one side of this little topographic high, this ridge, is really full of these sessile benthic organisms, these sponges and corals that anchor themselves to the rock. And then the other side of this ridge is much more barren. The bright yellow one you see just ahead, I'm being told, is the Elanthopasmia rostrata. Go ahead, Nav. Yep. Yep, that looks good. I think Bio wants to stay out of those chutes, and I think you guys do too. So I think just following these ridges is good.
Pilot, can we get a zoom just in the center of the screen, upper part of the screen? It looks like a little stick with one little branch. Looks kind of bare. Oh, okay. That's fine. It's a dead bamboo coral. I keep looking for carnivorous sponges. I'm kind of surprised that I haven't seen more. Looks like there must be an Iridogorgia down slope off to the left. I just see some little coil there. I guess that's what that is. It's quite a drop off right there. So a reminder, these slender, kind of smooth-looking whips, these are in the family Primnoidae, maybe the genus Convexella, the white ones. And then we're seeing lots of the orange kind of curly whips. Those are black coral, probably Aphanosticopathes. And then we see thicker, irregularly, I don't even want to call them coiled, but they're at the very least wiggly or curly. And those would be bamboo corals. And that's just showing you that those are three groups that are not closely related to each other. In fact, all of them have relatives, are more closely related to relatives that look like a fan or a bush that are highly branched. And it's telling you that evolutionarily adapting sort of colony morphology of the whip-like shape can occur many times throughout the tree of cnidarians anyway. It's interesting because as I'm saying that and thinking about it, you don't see that quite as often among plants, at least large plants. So cactus would be, some cactus are unbranched. I suppose if you think of like an ocotillo, they have, very long branching just from the base, but they don't branch after that. But most trees want to spread out their limbs and get their leaves into as much sunlight as possible. I think these frilly vase-like ones are the sponges that we collected a short time back. Got a nice piece of that. Lots of bamboo coral branching types and whip-like types. Lots of Chrysogorgia, these sort of bottle brush. They're almost peach in color. And then the ones that are a deeper brown orange, those are Acanella bamboo corals. There's some of the orange black coral whips that I referred to earlier. Seems strange to hear me say orange black coral. Is that on the rock, you mean? Huh. Yeah. So there's another, actually, can we look, just coming above center where the lasers would be, it's a sea star, but if you stay on the right side here, you might see the underside of it up above the lasers, almost at the top of the screen now. So that orange thing looks like it's got its arms curled backwards. So I think it's feeding on a Chrysogorgia, possibly. But the neat thing is, its mouth is pointing towards us, just like the urchin was there. Yeah, the laser's just pointing over it. Passed over it, excuse me. So this is a good angle to see if we can see the stomach everted onto the coral that it's feeding on. Oh, this is actually an Iridogorgia. Looks like it's feeding on an Iridogorgia. That's the spiraling coral. Very delicate branches. You'll see when we zoom in, you get a better look. So I'm trying to see, you know, are some of the branches, are there no polyps on them? And if not, that would be because this sea star has digested them already. And Chris Moss says this is the genus Evaplasoma, so a pretty well-known coral predator. And you can see all the bright red tube feet there kind of hanging on to different parts of the branch. I'm not actually seeing the stomach everted right now, so maybe it's taking a little break. Yeah. Maybe one lobe there on the right side looks, you know, significantly bigger than the tube feet. I suppose that's possible. All right, good stuff. So another example of sea stars feeding in the deep sea on coral. Okay, so I see there's some interest in one of the tubular sponges. I shall keep a lookout. Possibly Evaplasoma scorpio, Chris Moss says. So Scorpio would be the species name, Evaplasoma is the genus name. Another one of the Coreomorpha solitary hydroids. They're just passing out of the lower right. No, that's not for you, pilot. I'm just, just passing it on for sure. So one of the reasons that I call out a bunch of names is that you have various students on shore who are annotating the video. So all the video that you're watching is being recorded and becomes data for future analysis. And we have this marvelous tool that can be used through the Ocean Networks Canada website called CTube. And CTube allows scientists to add annotations that are time-stamped to correspond to what they're seeing in the video. And these get stored as a searchable data file that are also connected to all the metadata that's being recorded at the same time as the video. And by metadata, I mean things like what is the temperature of the water. Right here it is 4.1 degrees Celsius, for example. What is the salinity? How much oxygen in there? What's the exact position? And so sorry, I lost my train of thought how I started there. The students analyzing the video, the annotations. I'm also wearing a headset, listening to lots of different voices, reading the chat, and describing what I see. Oh, right. I was saying why I'm saying out all these names. So I say out these names so that later on, when the students are going back and doing some QA/QC of the video annotations, they can also reference what's being said on the video and relate that to what's being seen, because they're not necessarily experts in everything they're seeing, although they're becoming experts this summer as they see so much video. Lots and lots of these black coral whips starting to show up. There's another Metallogorgia colony next to a couple of black coral whips. It's kind of interesting how we go through these patches. Oh, have a look at this fish. Oh, that's one of the Oreos. The fall spore fish. Okay, we've seen that. That's the one with the producible mouth. So I was going to say, we go through patches. So you can think of patches on the scale of, well, this whole ridge that we're looking at compared to the whole mid-Atlantic rise, or this whole ridge compared to the whole North Atlantic Ocean. But we also have these smaller patches as we're moving up this ridge and as we're moving sort of west and east of it. And clearly there are some places that are more favorable than others for communities to develop. And it's going to be a function of, can the larvae get there? Is it appropriate to settle? Is the substrate stable over a long enough term that the colony can grow? And is there enough current moving by during that time delivering food so that they have the energy they need to grow? And so you have these micro changes in the environment where some areas or patches are more successful than others for any of the various reasons that I just listed.
I went down to the galley to get a cup of coffee, and I heard from the TV room that you were talking about C-tube. So I rushed in, and I was like, oh, we must be over rocks.
Oh.
Yeah.
You're pretty much right. We were.
Yeah.
Yeah. Sorry, Tab, that's the...
I'm a little surprised how far off we seem to be from the base surface. Yeah. Interesting.
40 meters, is that what I heard?
Yeah, that was what Chris said earlier.
There's another example of the parasitic zoanthid overgrowing a bubblegum coral. The bright yellow is the zoanthid and would be categorized as the parasite, and the red is the underlying bubblegum coral that's produced the structure that the zoanthid is overgrowing.
And they call it the bubblegum because of its color, not because of its chewiness.
Yeah, I don't know that anyone's chewed it, but you could get some characteristics there, right?
Yeah.
Taste, smell. Certainly people use smell.
Geologists are known for licking rocks to try and figure out the grain size for sedimentary rocks.
The boarfish hanging out there in the upper part of the screen, and something else, some other, I'm not sure if that's one of the, what were we seeing? Not the codling, but... So one of the things that the ROV is doing right now is just come up off the bottom, and it's spinning around, and there's a sonar mounted on the front of the ROV, so they can use that sonar to look for hard targets, so we know, what's the interesting part of this ridge feature to follow, so we don't have to just be out in blue water. Although there's a lot of interesting things to see in the blue water, as we saw in Dive 3. I'm seeing a couple of different Metallogorgia colonies up here now. That's a big coral back there.
I think it's mainly current-based. Yeah, probably these have been swept mainly clean of currents or overlying sediment.
You can turn over to camera 2. You'll get a view from Sirius of the D2 sort of spinning around on the top here, kind of looking out into the murky distance.
It's amazing how little sediment there is.
Using its sonar.
Which is an order of magnitude older than when we were in the axial volcanic region.
Mike Vecchione suggests we can refer to this mini school as a snack pack of Oreos.
I like it.
Yep. Go ahead, Nav. Copy that. Yep, that's great. We'll look for some midwater animals. So as Ashton said before, we've been sort of climbing this ridge, and there are some local highs, and then they dip down again, and then the high pops up again. And so, you know, I wouldn't exactly call it a roller coaster, but it is sort of moving up and down. And operationally, because we're always looking forward with our video camera, we try not to really travel downhill with the system. I mean, I guess you could turn around and back up down a hill, but it's not the preferred method. And so it may be, as we go forward here, we'll be in the blue water for just a little bit as we cross a short saddle before we reacquire the bottom and continue our way up. So we'll see what happens.
So I'm going to run some numbers past you, and I want to know your thoughts on them.
Okay.
All right, now these numbers are a little old. They're about a decade old.
All right. I really like 13. Is that one of them?
No, not yet, or 47.
Okay.
It's talking about epifauna, and it's talking about sessile and, is it vagal?
Vagil.
Vagil.
Yeah, so mobile.
Mobile and stay put epifauna.
Uh-huh.
And it says, with the 125,000 marine species of epifauna, this was 10 years ago, only 30,000 of them are vagal. Vagil.
Yeah.
Does that sound about right? Am I interpreting that correctly?
Boy, I have no idea about the numbers, and so I'm trying to understand exactly what's being said there. So.
Oh wait, here. Sorry, I misspoke.
All right.
Of the 125,000 species of epifauna, 30,000 species of infauna. Yeah.
30,000 are in fauna.
Are in fauna.
Yeah,
so 120. So maybe it was that.
That sounds a bit better, because otherwise you were saying
1 to 4.5.
they're both sessile and stationary and moving around.
Well, this is what happens when I use words that I'm just learning on the fly.
Yeah.
So
So what is that? Was that you said 120 and 30?
Yeah, 120
so about a quarter.
Yeah, about a quarter. And
so about a quarter of the species are burrowing into either a soft substrate or hard substrate.
Yeah.
Some are able to burrow into hard substrate, or maybe even like boring into the sponge, for example.
Yeah.
That's kind of interesting. I have no idea. Those are not numbers that I considered before.
No, so I guess I'm not trying to number call you out on general diversity, but I guess looking at these deep sea environments that we're looking at now, does that seem like it might hold true? I mean, of course, we can't see the ones we can't see.
Yep.
But do you have an idea of the diversity of the fauna versus burrowing versus living on these outcrops?
Yeah, so an interesting question for a couple of reasons, and I just want to point out, as we're sort of floating here, we're changing out pilots. That's why we're not moving very fast, but there was just a massive metallogorgia that we passed over. So the system that we're currently using is really good for seeing this sessile communities that are not moving around, and maybe the fish and some of the mobile stuff like that. It's not really good for understanding what's living in the sediment, what's boring into the rocks, what's hidden in the sponges, and so on. So I don't have a good sense of that from what I'm seeing here, because I'm missing that whole component of the community.
Yeah, we can't really observe what's beneath the sediment or in the cracks of the rocks.
Right, which is fascinating, right? So we can see today we've had tremendous diversity, but we know we're only scratching the surface, and maybe it sounds like from the numbers you just read, we're missing a quarter of what we could be seeing if we had the power to X-ray it, for example, or whatever.
Well, if I can take this hypothesis one further, you could be sampling for biology rocks and then just looking on the other side.
You trapped me.
And so you could just be collecting more rocks.
Entrapment.
Yeah.
I call entrapment for sure.
Not to be biased in any...
No. Well, as you know, I do closely examine every rock you bring up.
You do.
Yep.
You do. When the ROV comes back up, Scott and Arvin, our data science manager, rush out with with buckets, and they fill those buckets full of seawater that's from the ROV, that's been trapped either in the bio boxes or in the open containers of the ROV as it's coming up. And then they take the organisms that they have sampled from the deep, and they put them in those buckets of cold seawater, and that gives those organisms a longer time to be sampled and processed on ship. Now, at the same time, because the rocks I sample also usually have species living on them or attached to them, which we call associate samples, we also take those rocks and we keep them in nice cold baths of seawater. Now, it doesn't matter for the geology whether they remain cold and wet, but it matters for the biology. And so after Scott and Arvind process their biological samples, they'll then go to the rock samples and do an investigation to see whether there's any associate biologic organisms attached to those rocks. And then they'll remove those, process those, and then after that, I'll get a chance to actually take those rocks and clean them off, dry them, and then do a first description.
Yes, understood, pilot. And that was the sort of direction and overview we got from Chris earlier. And so are you essentially more or less following what you're seeing with your eyes rather than heading directly, for example, to waypoint 3? Is that correct? Got it. Okay, great. Okay. Make it so. Another pretty good sized Metallogorgia there, just coming into view on the left. The corals are leading the path. There's an even larger Metallogorgia on the farther left. That's our depth here, 1645. The sweet spot for Metallogorgia. It looks like there's a whole wall ahead that's just covered with stuff.
Mm-hmm.
Wow, look at that in the distance.
Yeah, so this is kind of similar to the maneuver we did when you were at the launch. We kind of came to this one of those outcrops and then skipped across the water column to the next.
It's kind of intimidating, looming up above us.
Yeah.
Yeah. I mean, I understand it's just the slope that I've been climbing, but I like to have a very small view of things. I get scared when it's a really big view.
So it's okay, Scott. I'm
Thank
here.
you. I appreciate that. Understood. Yeah, so we're going to cross this little saddle in the water for a little bit. And who knows, maybe we'll find something interesting in the water. Looks like we're passing an Oreo or false boarfish, Neositis, off there on the left. I think I can see the upper slope here.
Yeah, so that's the ridge.
Yeah. Let's turn around a little, yeah. We'll practice mid-water mode.
Turn around. Every now and then.
I think from the perspective of all the organisms down here, the ROV is definitely bright eyes.
Yeah.
Yeah. Maybe I can get.
Yeah, and so if you're just joining us now, we're just getting ready for a ship's move, where the ship will move, which then will move Sirius, which will then allow the ROV to move and have a different radius of maneuverability. And so we're on this kind of topographic high, this bathymetric high, this ridge. Oh, there's some beautiful rocks. There's some beautiful fragmented pillows. You can see the one in line right now. It has a rind with a little fracture down the left side of it. And then lots of broken talus, lots of pieces of rubble. And so one of the most surprising things to me whenever we dive in a new area is just trying to guess how much sediment there's going to be over the exposed rock. A few days ago, we dove right in the middle of the axial volcanic ridge, right in the middle of the mid-ocean ridge valley of the Mid-Atlantic Ridge, on an elongate composite volcano that probably couldn't have been more than 25 or 30,000 years old, in my estimate, and it was absolutely blanketed in marine sediments. And here, right now, we're on oceanic crust that's probably about 500,000 years old, and because of the ocean currents and the deep water currents, it's mainly free of sediment. All right, time to grab some rocks.
Yep, there's a shark on the top left. Coming in behind the Metallogorgia. Off it goes. Pilot, are you in a position where you could look at that large fan that's just downslope to the left? I understand it's downslope, it's a little tricky, but okay, thanks. I'm not sure if that is a bamboo coral like we've seen earlier or something else. The fact that it's so flat and planar suggests no. It could be just a really large Paramaricia, one of the plexaurid fans. And we collected a piece of that earlier. Yeah, I just want to get a look to, yep, exactly. It's looking more and more like a bamboo coral the closer I get. It's just interesting to me how planar it is. I wonder if it's a Jasonisis type.
That would be a J3?
Yes, very good. Nice. I got to learn some geology terms. Rock.
There you go.
Yep, there we go. I can see just a really thick stem down there. Oh, a nice view of a fish, too. Thick stem, I can see the nodes, and I can already tell from the polyps here that this is the J3. So thank you for that view, and feel free to take a look at the fish.
Les was asking if we can determine the direction of the current right now.
Yeah, and I'm seeing particles move from left to right, and our heading is 101 degrees, so just south of east.
So it looks like a
It'll be northern.
north to south.
Yeah, north to south current. So I think that's good for us and the fish, and since we only have about 40 minutes left, if you're going in the direction that you're pointed, you're not, okay. All right, no problem then. Okay.
So I sent some images and video from our dive on the axial volcanic ridge the other day to colleagues of mine, Deb Glickson, who's the geo lead for the next cruise, and then also David Clegg at the Monterey Bay Research Aquarium. Videos of the what I thought were very old extinct hydrothermal vents, and they both agreed that those were probably previously active hydrothermal vent systems. So we'll submit those to the InterRidge database. These active vents are extremely rare, one every 90 to 150 kilometers. So finding an active one is pretty interesting as well from a geological standpoint.
Yeah.
Would have been great if they were smoking, but
take what we can get.
Exactly.
Well, boarfish makes another appearance. So I'm reading a report that I got the other day from as an email. Oh, hold on. That's another boarfish. Several boarfish up here. So Ken Sulek sent me a message the other day where he was noting that super coral habitat is usually poor fish habitat. And the reason is that there's lots of rock for attachment for the corals and the sponges, but not much sediment. And the fishes that are associated with the soft sediment, which are most of the small to medium-sized microvores and mesovores that depend upon the invertebrates that are living in that sediment, or on the substrate surface, they would be missing. And there's only a few fishes that actually feed on corals or sponges in deep water, and the ones that he knows would be like these Oreos that you see swimming around that may be biting off the ends of corals, and also that Antigonia that we saw earlier. So that means that what we're left with when we're in a coral habitat like this are the larger megavores, those that cruise and search, hover and wait, or sit and wait for sporadic larger victim to appear, just like that shark that we saw a short time ago. And so then he goes on to say, you can't pack too many lions on the Serengeti, especially when likely small prey fishes, shrimps, and squids are in short supply.
It's a very timely email, given our just conversation about epifauna and infauna, and the lack of really knowing what the infauna is. And so I guess that's a telltale sign maybe that the lack of those populations of fishes might suggest that there's also a similar lack of infauna potentially.
Exactly, on a substrate like this. Yep.
Which is not surprising given we're on hard basalt and it doesn't seem to be very heavily sedimented in any area so far.
Les Watling points out that and this is based on some of the observations that we've made, that those Oreos might also be biting off the tips of the brittle star arms that are up on the corals. And very often you can see these brittle stars, their arms, that there's a clear disjunct position where the diameter of that arm changes, and it's clear that it's regrowing after it's been broken off or bitten off.
Yeah.
Brittle stars are well known for being able to, maybe I shouldn't say voluntarily drop their arms, but as a decoy, sever the arm. Basically, the arms are made up of what we call vertebral ossicles. So the ossicles are calcium carbonate elements that are embedded in the skin, and they form these vertebrae so that it allows those arms to be so flexible. That's why you see these brittle stars raise themselves up in their arms, and they're really twisty and bendy, unlike the sea stars. So they break at those vertebral points very easily.
Well, it reminds me of that old saying, don't bite the arm of the brittle star arm that feeds you.
Yeah, or do.
Or do, I guess, if you're really hungry.
Do bite the arm of the brittle star arm
if
that
you got
feeds you.
nothing else to eat.
Is this a ctenophore in the water column? Now I don't see it. It was going by really fast and it went underneath you. Yeah, no problem. This isn't a midwater dive. Just Chris Ma's noting that at least in the observations that we've been making over the last couple of days, we haven't seen brittle stars with those damaged arms. And so Les then says, well, perhaps we're misinterpreting, because we have certainly seen on the New England seamounts Oreos, and we've seen those brittle stars with the apparently regrowing arm tips. So maybe we're misplacing the accusation on Oreos. Maybe it's something else. Yes, absolutely. I'm not sure what it is. It's going pretty slowly. I'm kind of thinking it's a little salp, but I'm not certain. And I base that because I see like a little white dot in the center of it. Oh, it's a little siphonophore, I believe. Just like the tip of the rocket ship. It's the little float. So I'm not sure if that's just a really small colony or a colony that's beginning to develop, or if it's something else, but I know my midwater colleagues can tell me. Ken went on further to note that, these flat abyssal plains where there's a lot of sediment aren't really all that exciting to traverse with the ROV in terms of, constantly seeing new diversity and interesting things to look at. But they're really important and speciose in terms of, exactly as you said earlier, the infauna and the epifauna of the invertebrates. And so they're generally more abundantly populated by both benthos and the fishes that feed on them. He notes that shallow coral reefs are kind of an exception. There's a hard substrate community that has a lot of fishes, a lot of diversity, but it's a completely different habitat. The food availability is different with a lot of in situ production and so on. So it's not directly comparable to what we're looking at here.
Now, one thing that always impresses me from the geological standpoint is that, for the last 5 hours of this dive, we basically come up about 150 meters about in vertical relief, and we've seen all these kind of steep sheer walls and all this breccia and talus, and so about 150 meters. But that's only probably about a tenth of the thickness of the entire thickness of pillow basalts beneath us. So you can think about all these pillow basalts that erupted that form the upper portion of the oceanic crust are probably about 1.5 to 2 kilometers of pillow basalt deposits, of active eruption deposits, before you get to the harder, denser interior that was never erupted underwater.
That's amazing. So what's the time frame over which a ridge like this would build up? I mean, I know you told me the whole thing was perhaps on the order of a couple of million years old, but I got the sense that that's post-production.
Yeah, so the oceanic floor that we're on right now, right, which has been moved away from the active spreading axis, it's been faulted significantly, which is what we're seeing the exposed surface of. This is about 500,000 years old, 400,000 years old, and so you can imagine just over the time frame of maybe 1 million years, it moves about 1 kilometer off of that central portion, and probably within 250 meters. Maybe 250,000 years, it's done producing new eruptive products. So, over just a few 100,000 years time scale.
This builds up.
Yeah.
It's impressive. So this is the fish that Ken Sulek was saying has the protrusible mouth, and so probably that lower jaw is hinged and can move forward, and so better pluck things off of the coral, for example.
For the video crew out there, Les Watling was saying that there was a beautiful image of the ROV in the eyes of the Oreo, in the reflection.
Ah. Oreo dreaming of ROV.
Yeah.
I see a haiku in there somewhere.
Oreo dreaming. ROV. Oh, could it be? Fly away, explorer.
That's a sad one. That made me sad.
Oh.
That's good. I like that. That's a good shot. Okay, now you got me wanting to look for my Lonely in the Sea Snow poem.
Oh.
I mean, if this is poetry hour.
Oh,
Look at this. What's going on here? Looks like somebody's dug in here already before.
Yeah, that's really heavily sedimented. And,
well, doesn't it look like a gouge?
Yeah, I wonder, it could be.
Huh.
I mean, you were talking about rock dredging earlier, right? So you never know.
Yeah.
I would have expected maybe a longer drag, but sometimes when they're dredging for rocks, they aim for kind of the tops of steep escarpments because they know they're going to kind of hook those teeth of that rock dredge in. But it's definitely been overturned and it's much more filled with rubble.
So you have to keep that one written down. Here's the one that I did. It's very similar thematically to your more simple
it
haiku.
Is. I thought that might have gone in a different direction about sampling for a second. That's sad.
That would be really sad.
Is that something
Can
you
we
wrote?
get a close-up look at the same way you did just now? Can we get a close-up look at these globular sponges on this coral, please? There's 3 of them. I see that I think all 3 of these little balls, sponge balls, are the same, but the one that was on the upper right, I could clearly see some chimney-like oscilla excurrents. That's a good view. Yeah, this is a nice view of the surface. Oh, look at that. So before you turn over, pilot, so these crossing swords, if you will, those are carnivorous sponges. So finally we find some carnivorous sponges. The sticks that have these, what appear to be spines sticking out of them, I believe they call them filaments. This is a kind of sponge, and there's a group of sponges that evolved, a clade. I believe it's now one family. It's quite speciose, so keep finding new examples. But they have learned a whole new way to feed in the deep sea. And there might be an example of that. Let's see. So you see 2 of them are forming an X, the 2 main ones. And then if you go a little bit lower on the right, where we just did the focus, there's another X. And to the left of that X, there's something on the stick. And basically, these guys have their spicules that are kind of, they're described like Velcro. They're hooked. And they entrap zooplankton. And then the cells are able to migrate and basically enclose the zooplankton. And they're also enclosing bacteria. The bacteria then digest the zooplankton, and the sponge takes up the products. Pretty crazy. Yep. Yep. I got that earlier. I'm not sure that these are big enough. We'd have to pull up that whole piece of coral. These things are tiny. Likely the genus Asbestopluma. I see some caprellid amphipods in the sponge. So I think all those little chimneys you see are the excurrent openings for this sponge. So, pilot, there's a lot of interest in a collection of a variety of things here. So let's just pull out a bit and look at this. So I'm wondering, let's see how this is oriented. So you notice what looks kind of like a thick branch of a tree that's running across there, and that globular sponge is sitting on the end of it, and I think the carnivorous sponges were also sitting on it. Yeah, it's the whole. The point is to get all of them, and I don't know if Chris is still up there. He knows how hard this is going to be. Once again, they're all growing on this really brittle coral, such that when you try to grab this thing, it's going to crumble around you. So I'm not sure how to approach this again. And I think the best would be ideally if you were on the other side of it looking at it, but I don't think we can get the... That's what I meant. Yeah. Yeah, I don't think you can do that either. Yeah, then you're going to have... Yeah, I think it'd be worse because then you'd have the left part of that, the harder colony, sticking up in front of what it is that we really want. Can we have the lasers on just to see what's the size of this? 10 centimeters. So that's relatively small. So I wonder if you brought the claw in from the right side and just sort of tried to grab below the globular sponge that's just above the left laser dot. Do you think you would get everything above it? Yeah. It's horizontal. No, that's not what I mean. What I mean is the thicker, it looks like a thicker bare branch that goes horizontal, and the globular sponge on the upper right is sitting on it. Yeah. But those crossing swords are also a target. So it's kind of like we want that whole branch, but of course it's tucked up against the thing that's going up to the left. That's the challenge. That's why I say maybe if we try to get below it, but... Huh. I hadn't thought of that. I didn't think it was that small, but if you think it's that small, sure. Okay. If not, the only other thing I see is you bring the claw down from above right over those crossing filaments, and then we just take our chance and hope we get it. Can you zoom in again? And yeah. So zoom in on that large ball in the center. Yep, that's the branch I mean. So to the left of that dot, you see those two filaments, white filaments that are crossing? Exactly. So I'm hoping to get those in. So I thought if you put the claw in coming down vertically and grab that branch and just broke it, you would get both the crossing things and the sponge that's on the end of it. That's what I'm saying. You're going to mangle everything. It's yep. I just don't see how you can get in there without taking everything, which is fine. You can do that. Yeah, just grab a handful and see what we get. That sounds good. Yeah, go for it. Oh, yeah. I didn't even notice this one. It's interesting that you say that, Brian, because this was also a target earlier. Yeah, I don't. Yeah, sorry. Apologies. I don't have a good sense of that because I can't even see where the carnivorous sponge are actually arising. I'm sort of guessing that they're coming off of that main branch. They kind of sort of disappear behind the back, you know, so it's hard to say. But it makes most sense that they're attached to that same point. Yeah. It sure looks like they're coming off of that, again, that horizontal branch. But all this stuff is interconnected, so it's tough. You know, and it's even possible that when you try to section this, that would be enough to make it fall. And if that's the case, then maybe we can just pick up what falls. Yeah, let's see what we can do. This is a really tough one, you know. It's a tough challenge. Yeah, well, that's why I think you should grab a handful, and we get what we get. Okay. Let's grab a handful, come in from the right side, and let's just see. Yeah, it's kind of like a good luck thing. Take a handful, and we'll see what we got. And do not worry when you see stuff tumbling. All right, you got the globular sponge, and you got part of that branch. And you got the tubular sponge. That's terrific. Okay. I'm watch lead 2, right? Okay. No, one's got to be an associate of the other, I think, because they all come from the same time. And I'm getting a name for the first one. Right now, I've asked for an ID, which I don't have, so right now I'm just calling it globular sponge. All right, terrific. Okay, so very nicely done, pilot. On shore, we got those 2 samples are stowed in the bile box. So we got at least 2 of those sponges, the globular one and the tubular one, and then whatever associates were on the part of the branch that we managed to get in the bile box. So, and if we're lucky, there will also be a carnivorous sponge on there, and I will look at it closely. Good stuff. Still want your rock? There's still time.
No.
Oh, there's a metallic gorgia there on the right side, and it must be missing its tissue going down the stem because I saw a whole bunch of associates overgrowing it. Yeah, I think we can keep going. Thank you. Maybe this iridogorgia, that's the spiraling colony just on the right side. If you can get the lasers on it, that would be good. That might help us put it to a species. That's great. Nope. No, that's good. I think that's Magni spiralis or Magnus. I think is it Magna or Magni? One of the two. I think I was so excited earlier with all of the density and diversity that we were seeing, and I didn't take the opportunity to take an eDNA sample while we were in the midst of that community, which is a real shame. We did get one when we first landed on the bottom. There's a little bit higher. There we go. We're getting to another little high point here that looks like it's got a lot of stuff on it. So, Pilot, as you get above where you see all these corals and sponges growing, let's trip one of the Niskins. Thank you. What's that? Within a minute. Yeah, let's maybe go 2 more meters forward. Thank you. What I'm trying to do is get the back of D2 over this feature that I had the corals and sponges on. Thanks. Copy that. I'm ready to go. Sample taken. Thank you, pilots. Pilot, if we can zoom on just to the right of center, there's a coral, black coral. It's the bushy thing that's got two pink dots on it. So almost in the center of the screen, just right of center, near the top of the screen. Yep, it's like 2 o'clock to the lasers. Yep, now your lasers are right next to it. Turn right. And what I'm looking at is there's something in the branches, the upper branches, that's a soft pink color, just above the lasers now. Yep. Thank you. Understood. This will tell me if it's something worthy of closer inspection. Is that an urchin that somehow climbed up there? That is one talented urchin. Huh, fascinating. So look at that. That urchin somehow has managed to get himself up there with all the spines, and notice all the skeleton below it is cleaned off black, but above it is this orange tissue. So I think that may be feeding its way up this black coral, perhaps. Can't guarantee it, of course, but it is a smoking gun. There's also a couple of brittle stars on there, and there's a crinoid. This is a Leopathes black coral, if I didn't say that already. I think Chris Ma is agreeing with me. Chris, we are trying to get a closer look at the urchin. He's saying this is not Okeanos. So earlier we saw a great big urchin, kind of pink in color. Didn't have spines quite this long. He said that was Okeanos. This looks like something different. Chris, we're at the end of the tether right now, so D2 can't get any closer, and that's why, we're not getting quite as good a shot. Huh, that shrimp is carrying something. That was interesting. Oh, he's gone. Eagerly going off home to feed on whatever he just picked up. So I think we're getting some information passed on from Tina that she may have seen a second urchin on the lower base on the stem, but also that Leopathes often have a naked stem. But I thought I saw lots of the internal branches there were also stripped of tissue. Oh, yeah, I see the second urchin now. It's on the lower cleared part of the branch. Yep.
Sorry, I'm just joining. How much bottom time do we have left?
I think 15 minutes, approximately.
Thank you.
0 minutes? Okay. Yeah,
0
so this
minutes?
is, yeah. 0. Yep. Les Watling was noting that at this depth we would have expected to see some coralliids, the precious pink corals, and we didn't see any of those today. We did see one Candidella, but not more, and we just saw the convex cella whip. That means that family Primnoidae, which is usually so abundant, we didn't see. Let's go ahead and do one more, trigger one more Niskin once you're stable here. Not stable, but you feel like we can do that. Stand by. All right, so just doing a little more paperwork there with another eDNA sample, water sample. So thanks, everybody. So that's the end of the seafloor portion of the dive. We're going to spend the next hour rising up through the water column. So if you would like to watch the blue water and hope that some squid swim by, please stay with us. For those of you on shore, scientists who would like to participate in the dive planning call for tomorrow, we're going to do that in just a few minutes. I do need to check my email message, make sure I'm not leaving anybody out, but I do think I said 3:30. Yes, 3:30. So just give us a couple of minutes to get set up in here with the mapping team and the expedition coordinator, Derek. I think it was a really interesting dive today. We came down in an area that had a very high density and diversity of corals and sponges and associates. We didn't see a huge diversity of fish or abundance, but that was something expected as I relayed the information from Ken Sulak. Yes, Mike, sorry, just giving you a teaser there. But I personally thought that this was a really exciting site. I'm really glad we saw it. I personally have never seen such an abundance and diversity of corals in the North Atlantic, so at least at this depth. So that was quite exciting. So stand by, everybody on shore. Thanks for those of you who participated throughout the day, providing us with some information. And we'll get on the dive planning in just a couple of minutes, and then we'll be diving again tomorrow. Same time, same bat channel.